loci just means more than one locus. And then what's a gene? A gene is a DNA sequence that encodes a protein or an RNA. So not every sequence is a gene. Some sequences do not encode proteins or RNA, that are transcribed. So it's very specific kind of sequence. And then what's an allele? Allele is a version of a DNA sequence at a locus. So an allele is just, it can be one nucleotide, like one SNP, or it can be a combination of different nucleotides. So, but it's just a form of a sequence. So either combination of nucleotides or single nucleotides, or it can be even structural, so presence or absence, insertion deletions. So it's just a version of a sequence. And then a genotype is a combination of two alleles. Allele is just on one chromosome strand. Genotype is a combination of two alleles. And then segregation refers to separation of two alleles during reproduction. During sexual reproduction, when meiosis occurs, alleles are separated to different gametes. They get inherited separately. So we do not inherit genotypes, inherit alleles. The genotypes are separated. Often we say something is segregating in a population, like a trait, for instance, is, oh, this trait is segregating. It just means that we see visible variation in the progeny. So if you see have a couple of parents, cross them, generate progeny, you can say, okay, we see this certain trait is segregating. It just means that we can see that there were two alleles that separated, and now they're being inherited separately in the progeny. And then population is pretty broadly defined, but it's a group of individuals that can intermate. So in this kind of a breeding definition, but there's a lot of different kinds of populations. So, but population in a population genetic sense, it's a group of individuals that can intermate, and they share common ancestry, so they can trace back to a common set of founders. Okay. And everything kind of is rooted in this Mendelian model of inheritance. So it just says that traits can be explained by single genes. So major ones like seed shattering, maybe familiar to you, pea color, from Mendel's experiments. These are just say, okay, traits can be explained by single genes. And then there's the three laws, right? Dominance, segregation, independent assortment. Dominance says that there are some alleles that can be dominant over others, so that just having one copy is enough to confer the phenotype of, so if something is in heterozygous or homozygous state, they'll have the same phenotype. Segregation, that's really critical, meaning that the alleles separate, they're inherited separately from parent to progeny due to meiosis. And independent assortment, meaning that if you've got if you've got alleles at different loci, those are also inherited separately. Now, that's kind of nice and neat and simple, seemingly simple. But the problem with that is, or at least at the time when Mendel was trying to propose this, this model of inheritance, it didn't seem to explain continuous variation. But how do you explain, variation in human height, for example? That was always the example that everybody gave to say, how can this model of inheritance possibly work if you've got these traits with continuous variation? It's not like a pea color, green and yellow or something. It's just hard to explain. And actually, that's where the infinitesimal model of inheritance comes in. So it reconciles that continuous variation with the Mendelian model of inheritance. It says that traits can be explained by an impossibly large number of unlinked additive alleles, like human height, skin color, for example. And each one behaves in a Mendelian fashion. So the same Mendel's laws apply to each individual locus, but you have a lot of them contributing to the trait. And each one explains a very tiny amount of the variation. And this, in this infinitesimal model, of course, it's saying that there is an infinite number of loci. So that means that if you think about infinity, each individual locus practically has zero effect, almost zero, close to zero, because, you know, that's how it works. You can't, if you have an infinite number of something, each one has to explain an infinitely small amount of the variability. Now, of course, not everything perfectly fits that, because we know we can't have infinite loci. I mean, genomes are finite, so you can't have infinite alleles, but there's so many that practically fits, it goes to infinity. So it fits this pretty well. And then this model even works when the number of alleles is rather small, even, you know, kind of not that close to infinity. Even if you have, you know, 10, 15 alleles, it starts to fit this infinitesimal model. So here's one example with here with skin color. And this is just, it can be any trait really, but this is just a textbook example. And you've got three alleles that, or three loci, sorry, that affect this trait. So you've got A, B, and C. So let's say the small a, small b, and small c alleles are with less pigment, and the big A, big B, big C, more pigment, right? So that's kind of two contrasting phenotypes. You've got alleles in both directions and only three separate loci. With three, you might think, like, how can this generate, how can you generate so much variation with only three? And this figure kind of explains it. So you've got, let's say you've got these two parents with contrasting phenotypes and genotypes at just three loci. Then you've got the F1 generation where everything is homozygous. And there's no variability, of course, since this is like your F1 hybrid. And then those, you know, self-pollinate or intermate, and then you generate the progeny population, which is segregating for all these at these three loci. And you can see that just by all the combinations, so this is every square is all the possible combinations of alleles at these three loci. And you can see all the variability that's created just by having that those different combinations at just three loci. So you've got extreme, you know, the parental phenotypes are even here. So you've got these, all the small alleles, all the big alleles, and then you've got different combinations. And so basically then it turns out the more of the dark alleles that each of the progeny has, then, you know, the darker the pigment in the skin. So that's, you know, that's just an example showing that you've got at just three loci, you can generate all these different combinations and get all that different phenotypic variability. And then if you map this out, you know, if you look at just the distribution of phenotypes that result from this, it fits a normal distribution. So you've got the majority are in the middle, and then you've got, you know, you've got fewer at the more extreme phenotypes. Now, What are some of the assumptions of the infinitesimal model? Of course, that there's an infinite number of unlinked loci that affect the traits, and each one has a very small effect by default. If there's a large number of loci that affect the trait, each one has to have a very small effect. It also says each locus has the same effects and frequencies, so each one kind of contributes equally. And that kind of makes sense if you think about each one having a very, very tiny contribution, then they're pretty much influencing the trait to the same degree, very, very small amount. The other assumption is that selection will not affect the frequencies nor the genetic variance. And that makes sense because if each individual allele has a tiny, tiny effect, then selection is not going to see individual alleles. It's not going to affect those frequencies because practically each allele has almost zero effect. It's kind of hard to wrap your mind around because it's like you're saying everything affects the phenotype and nothing affects the phenotype. So, but the selection will not affect allele frequencies nor genetic variance because each one is having such a tiny effect. And we know that this is not completely true because we do actually, selection is effective at, it does affect allele frequencies and genetic variance, but it just doesn't affect it very much. So it's just a small amount. And the more that the trait behaves like, has a large number of loci affecting it, the more that this model will hold true. Okay, so most traits that are important to us, I shouldn't say that because there are a lot of traits that are, I guess, not fitting this, but a lot of traits that we care about, especially in agriculture, fit this infinitesimal model. So, for example, yield, quality also in some cases fits this model. And if you think about traits that have been under selection, like directional selection for a long period of time, then if there were large effect loci affecting the trait, they probably would have gotten fixed already. So you can think about it. If directional selection has played a role in the evolution of that trait in that population, more than likely it's going to behave like according to this infinitesimal model. So traits that where let's say like in some cases one allele is favorable, in some cases another allele is favorable, that's where you can start to see more larger effect alleles segregating, and maybe this model doesn't fit as well. But traits like, especially thinking about yield, all those large effect loci probably got fixed. Like shattering would have been a big one that during domestication got fixed because it would have such a huge effect on yield. Okay. Okay, so now we just covered models of inheritance, so Mendelian, infinitesimal. Now we'll talk a little bit about allele effects. So basically they fall into two different categories. We've got additive, where an allele effect does not depend on other allele states. So it doesn't matter what the other alleles are doing, it's just the effect due to that having just that allele on its own. And then there's non-additive, where the allele effect depends on other allele states. So a couple of non-additive types of effects include dominance and epistasis. So dominance, that's non-additive because it depends on the presence of it depends on what other allele is present at the same locus. And then epistatic, the effect depends on an allele, if an allele at another locus is present. So let's see if I can show you. So a good way to illustrate additivity at a locus, if you can think of, you've got, let's say you've got a locus where you've got different genotypes, AA, this is A, and AA. So you've got these three different genotypes at a locus, and we're just going to look at the additive effect of this allele, A, big A. So let's just map this out, where you've got these three different genotypes, AA, and AA, and then you've got the phenotype here. So say you have a population and you look at the phenotype distribution at individuals with these different genotypes. So we can think about it like a box plot. So let's say this group of individuals with small a has this phenotypic distribution. And then the group of individuals that are heterozygous, so big A, small a, it has this distribution of phenotypes. And then you have big A, you have this distribution of phenotypes. So if you look at this now, you can see there's a linear trend where as we increase the number of big A alleles, we increase the phenotype. So we can actually draw a line through here. So there's this this is what we mean by an additive effect. So there's an additive effect of, so every time you add one more big A allele, you get that increase in phenotype. And so the additive effect is just the slope of this line. And that just means, okay, so for every unit increase, for every one allele that we add, how much do we increase the phenotype? That's the additive effect. Let me say alpha. Additive. For every added, how much now, if we did this for the opposite allele, it would be the same, but just in the opposite direction. So we could just rearrange the x-axis so that, and then it would just be, the line would just be going down. So it doesn't really matter which allele you take as the reference. The additive effect is the same, just changes the direction. Okay. All right. I think I will also explain dominance before we move on to that. So, okay. So dominance would look like something similar. So let's say we've got AA, and AA, and then we have the phenotype. And then again, this is the distribution of individuals, their phenotypes with this genotype. And then the homozygote, actually it looks like this. And then the heterozygous big A, it looks like this. So that's just showing you that, okay, this group of individuals that's heterozygous, so it only has one big A allele. It has the same phenotypic distribution as the group of individuals with two big A alleles. So this means this is an example with complete dominance. Now, what's interesting is there's still some additive effect that explains this trend. So we can try to draw a line through here. It's not going to fit perfectly, but it still explains a lot of this variation. So you can still estimate the additive effect at this locus. And now you've got a component. So now you've got some additive effect, and then you have some deviation from additivity. So you've got, these deviations from the line. It's what we call dominance. Dominance deviation. So we still have an additive, there's still an additive effect here. Adding more big A alleles increases the phenotype, but it's not a perfectly additive effect. There's some dominance there. And so that's causing this, what we actually observe to deviate from this additive model. But still, additivity explains a lot. There's just this dominance deviation. So there's the same way, the same thing can apply to epistasis as well. So you've got additive effects, and then you have deviations due to non-additive effects. But we always consider additive to be the main component that explains the variability. So, and then everything else that is not fitting an additive model is like a deviation. It's something that doesn't perfectly fit additivity, so it's sort of whatever is left. It's kind of goes in a sort of a residual. So you might be thinking, okay, well, why are we so obsessed with additivity? Why do we want to explain everything with an additive model? And the reason for that is just by thinking about breeding value. What's the definition of breeding value and what actually gets inherited from parent to progeny? Now, the additive effect, so breeding value is just the additive genotypic effect at a single locus or summed across multiple loci. So it's, if you take each individual locus and determine the additive effect of each individual allele and just sum across the entire genome. I think I have a couple examples so we can look more closely at that. And that's the effect, that's the genotypic effect that is passed on to the progeny upon mating. So only individual alleles are passed on to progeny from an individual, right? So we don't really get to contribute combinations. We have meiosis that happens, recombines different alleles. Also, we only get to pass on, one of our two alleles at every locus. And so that's why, just these additive effects are considered part of the breeding value. You only get to contribute, additive allele effects to progeny. And so that's really important for selecting parents. It tells us the expected genetic value of the progeny. So if you take two parents, basically the progeny that two parents generate will have, the average value of genetic value of the progeny is the average breeding value of the parents. And so the breeding value determines how good the progeny are going to be. You can have a really, really good parent, and maybe it's good because it has some specific combinations of alleles and so on. Once that gets disrupted, that can't be, by meiosis, it won't be passed to progeny. And so that's why breeding value is important. All those additive effects can be great, but if you're going to use it as a parent, you want something that has a lot of good additive effects that can be passed to progeny. Okay. So just to kind of illustrate better, breeding value of a genotype, at a single locus and across multiple loci. Here I give you an example. So let's say we've got, I'm just going to draw the what I illustrated before. Okay, so let's say we've got this allele AA, and we've got these phenotypes. Okay. There is something called the coded genotype. So this is just, if you take those genotypes and then recode this, instead of calling them with letters, we count the dosage. So we say, what's the dosage of the big A? So in this case, it's 0, and then it's 1, and then it's 2. So we're just recoding from a letter system to dosage of one of the alleles. And then we can recode it, we can center it. So basically just subtract. So now we just subtract 1 minus 1. And then we will have -1, 0, and 1. Okay, so then we have our coded genotype. Let's say we have our coded genotype is 1. And we say that this additive allele substitution effect here is 0.001, just as an example. That would be the slope here, alpha. We would just multiply alpha times 1. So this is the effect times the genotype. So we've got 0.001, the slope of that line, times 1. And that just gives you exactly what's on this line here. So we're referring to these coded genotypes. So it's just the slope times the genotype in terms of dosage. So that gives us, so, this is maybe seems complicated, but all you need to think about is think about the graph and think about the dots on the line. So the points that actually fit the line, where they lay on the line, that's going to be the breeding value of that genotype. I'll give you another example. So let's say we had the dominance example, or something that doesn't perfectly fit the additive. So we've got AA, AA, AA. This is phenotype. So if I count, I just say, what's the dosage of big A? It's going to be 0. So this is dosage 0, 1, 2, and then centered dosage. It's going to be -1, 0, and 1. And then let's say we've got these phenotypes. And this time it's not perfectly additive, doesn't perfectly fit the additive model. But let's draw the line here. Now look at the points on the line. So the breeding value of this genotype at this locus is going to be the value here. This is breeding value of AA at this locus. And so on with all the other genotypes. And so the breeding value is just this point on, this point on the line that is going through these phenotypes to try to explain them with an additive model. Let's see, let's see, I might have another way to explain it. Yeah. Okay. So now, individuals don't just have one locus. Of course, they have there's a lot of different loci in the genome that affect the trait. So if you look at a multi-locus genotype or an individual that has multiple loci, looking at across all the different loci in the whole genome, like what's the breeding value of that combination or that individual? You'll sum across all the single locus breeding values. So if you've got 2 loci and you have the breeding value of that genotype at that locus, then to get the combined breeding value, you just take the sum. So it's just looking at each individual genotype at each individual locus, what's the breeding value of that genotype, and then sum across the whole genome. So that's how we actually get to the breeding value of an individual, which is a combination of a lot of different genotypes. So then you get total breeding value. So in this case, let's say we've got, this is the additive effect here. And then we've got the genotype recoded and centered. So this is additive effect times the genotype. That gives us the breeding value at locus X. Locus Y, we have the additive effect. This is just made up numbers to illustrate. So this is the additive effect of an allele at locus Y times the genotype and recoded and centered. So that's -1. And then to get the total breeding value, you just take the sum of these values. So this is the breeding value at locus X, and this is at Y, and then this would be the total breeding value. So if this was an individual that had only 2 loci, this would be its breeding value. So it's just the sum across all the individual loci. And I think I explained this a little bit already, but it's worth emphasizing that dominance and epistasis are usually expressed as deviations from additivity. So we refer to them as deviations. So they're kind of whatever is left over after we cannot explain it with an additive model. We can try to explain it with dominance or with epistasis. And even a completely dominant locus, an additive model explains a large part of the variation. And so it's like, sometimes it's surprising to people how well an additive model fits, even though it's like, we know that nothing can be perfectly additive, like nothing will perfectly fit the model, but because it does fit, the large component of most effects, it adds up and it tends to explain a large proportion of variation. So that's the end for lesson 1. There's probably a lot of questions in the chat, but I haven't been able to read them. Let's see. So somebody asked about vegetatively propagated crops where dominance and epistasis can be fixed. So that brings up a good point. When you're looking at something that's like a clone or even a fixed line, of course, dominance and epistasis, they are important for the overall performance. But if you're going to if you're going to cross something and generate progeny through sexual reproduction, you will always disrupt those combinations to some extent. Of course, there's going to be some combinations don't get disrupted, so you will keep some. So it's not 100% true to say that combinations are not at all inherited. I mean, they kind of are, but we should assume that combinations get disrupted during meiosis. So okay. And some are asking about recording, and we are recording right now. So yeah, okay. I think I got, okay, what other questions do we have? Yes. Yes. Yeah.
So if breeding value is the coded genotype times the effect, is the heterozygous always having a breeding value of 0?
Yes. Yes. Yeah. Yeah, so they should be basically once it's recoded, what it will look like. So this one should be 0, and this one should be positive alpha, and this one should be negative alpha.
Chat stuff right now.
Oh, sorry. Yeah. Here, let me draw this in black. Yeah.
And would you transform your phenotype to just be between 0 and 1?
No, so it doesn't matter, like your phenotype can be as it is. And all these different effects, once they add up and they'll generate a realistic looking phenotype. So because you'll have, let's say you've got, one locus. So let's see, if at one locus you have like big A, small big, so this one gets to be 0. Let's say you've got VB and capital 1's always positive, let's say, then this one will be, something 0.05, some number, and then next one, CC, it will be negative 0.01. These are just made up numbers, but you can see across all the different loci, you'll have these values for, what's the breeding value of that genotype. And then you will just add up across all the loci. And then that gives you the total breeding value. Yeah.
I think there were a lot of questions about negative breeding values, allele doses. When you got -1, 0, 1 versus 0, 1, 2. So what is exactly that mean?
Right.
Nomenclature versus
so it doesn't matter all that much, honestly, like whether it's centered or not. I think there are some advantages in interpreting effects later on, like there's some advantages, but yeah, it's kind of, well, is that true? I think you can still have negative breeding values even if it's not centered. So because breeding value, so let's say breeding value of an individual, like you have a population and you've got, you're going to have an average breeding value. So now we're just talking about individuals and their breeding values. You have an average breeding value, and let's say, it just depends what you're consider. So usually usually we're talking, usually breeding value is always looking at a population like within it. So you're comparing individuals with each other. And so there is an average. Typically, we assume like the average is 0. Often we assume average for the population breeding value is 0. And so there's going to be breeding values above and below 0. So it doesn't really matter though what we say. Like we can say this average breeding value is 0. And then you've got high breeding values, and then you've got low breeding values. Okay. Now, let's say we say, okay, well, my average is not 0, it's 10. Okay. So we can add 10 to everything. That doesn't mean that doesn't change anything. You just add a constant. So the average of the breeding values doesn't matter much. It matters just individuals relative to each other. So there's different there's different assumptions we can make. Like we can assume that like within a population, average is 0, or we can assume that some founder population like years ago, when we first started the breeding program, that's 0. Those are having 0 breeding value. And then everything that we have improved is relative to those founders. So that's those two assumptions we can make.
So how do you actually parse out that this is the effect of a single locus, right? So, we have AA, this locus, but what if it's some other locus that's making the phenotype increase there?
Right, like there's basically confounding
Yeah.
because we don't get to observe them like individually. So what we need is a population where there is segregation at that locus. And then we kind of assume that the genome is like otherwise randomized. Like there's variability, but it's kind of occurring at random. And the population, so that like the rest of the genome has variation, but it's not dependent on the allele that we're interested in. So
you need to have a ton of individuals, pretty much.
A lot of individuals and a lot of recombination. That's where we can get the best like estimate of allele effects that's more consistent. But I don't know, probably when we talk about, maybe on tomorrow is Alex's, you're talking about that, I think, right? About how to estimate allele effects accurately, like even because there's going to be some confounding with your locus of interest and like other loci.
Yeah. I go over that stuff, but maybe not that. Maybe from a slightly different perspective. But,
Yeah.
But basically, yes.
Yeah. So
There is a review paper by Joe Gage where he talks about estimating association map populations. One of their main benefits is their ability to actually estimate the genetic effects versus like other kind of population out there. We have to put the name.
yeah. I guess another thing to keep in mind is it's going to be context dependent. Like your estimates of allele effects are going to be within a population and within a set of environments. And so it's not going to be the case where you can say like that you're not going to ever be able to estimate an additive effect that is like universal. It can just be like universally true. Like it's true within that context. So yeah. Like if there's 3 different alleles. That's a good question. So I think it's, you can still probably break it down into dosage, or just do presence absence of each individual allele. Yeah, I guess it depends if it's, are you talking about like a triploid or just 3 different
Yeah,
alleles?
very good.
Okay, yeah, then, let's say you've got, so I don't know, so 3 different combinations. Okay, so let's say these are your 3 alleles, A, and B, at one same locus. Then it's like, you would have one of these slopes for like presence absence. You would say, okay, there's a big A or there is no big A, and then you would generate different genotypes. So you can say minus, so there's no A or there is one, and then same for small a. There is none or there's a small a, or there is none and there's a B. And then I think it's the same. You've got, so this would be, oops, what happened? Uh-oh. Sorry. So you'd have, this would be 0 and this would be 1. So if you recode it again, it'd be 0, 1, 0, 1, and 0, 1. Yeah. Let's check the chat again. I think we can. He's asking about the volume. Yeah, someone's asking about that. Someone's asking about centered dosage and dosage. So I think I'll just briefly explain again, but centered, the dosage just is exactly what it means. It's like counting, number of alleles. So you can have 0, 1, or 2. Centering just subtracts 1, so that having 1 copy is actually, now we consider that 0, so that it's centered, so that if you take the average, like you have -1, 0, 1, it's centered at 0. Okay, so now should I take a 5-minute break, 10-minute break? I forgot. When did we start? It's only been 50 minutes. Maybe I'll go longer. I don't know.
I do have one more question.
Yeah.
Is the breeding value of a heterozygote 0 because you can't predict what's going to be passed on to the progeny? Since it's the measure of genotypic effect that's passed on to progeny.
So it's just 0 because we made it 0.
Okay.
But it's convenient in that way that, yeah, it's
If you could
intuitive.
predict that from, like double homozygous dominant or
Yeah,
big A, then you would be able to predict what's going to be passed on.
right. And you can think like if you had, so another maybe intuitive way is like you've got a heterozygote and one of the alleles is unfavorable and one of the alleles is favorable, and so they cancel out and they make 0. Yeah. Okay. So now let's see, we're going to the next lesson. We're talking about phenotypes, genotypes, and breeding values. All right, now this is where we make the connection between genotypes and phenotypes. So we just talked about additive genetic value, that's breeding value. how do we get a phenotype from there? We also want to understand phenotypes, breeding values at the population level. Okay, so this is really critical. It's just the definition of what is a phenotype actually. So you've got phenotypes. It's the sum total of environment, genetics, and then some residual. So environment is kind of a common effect, right? So we've got a bunch of individuals in an environment and they share the same environment. Everything is affected in the same way. So environment in this case is kind of a constant. And then the genetics, that's all the in this case, we're talking about additive genetic value. So basically breeding value, all the things that are heritable. And then you've got the residual. That's all the things that we can't explain. It's not specific, it's not like a constant from the environment, and it's not heritable. So these are just can be just random error, just noise, whatever cannot be explained. Okay, and then genetic value, we can break it down into components. We were just talking all about breeding value. So, but if we think about total genetic value, there's actually, breeding value is a part of that, but there can be other parts of genetic value. So let's see, genetic value. Okay. Actually, I'm going to go back one more. So this one, sorry. So we have phenotype is the sum total of environment, genetics, and residual. Genetics is, so you have total genetic value equals breeding value plus non-additive genetic value. Okay. So even the genetic component, we can break it down further. All right, so I just did that from the previous slide. Okay, and then breeding value is that portion of the genetic value that's transmitted for all the reasons that we just described. It's the portion that gets inherited. It's only the additive effects, and it tells us the value of an individual as a parent. And sum total of all the additive effects of that individual's alleles. So breeding value is just one small part of phenotype. So phenotype, you got environment, genetic value, residual, and then of genetic value, breeding value is a component of that. So it's just one component of that explains phenotype. So we're interested in predicting breeding value based on phenotypes. We want to know if we're plant breeders and we want to improve a population, we want to know what are the breeding values of our individuals so that we can select parents. If we want to just release varieties, we don't need to worry that much about specifically breeding value. We want to know total genetic value. But if we want to select parents, we want to identify individuals that are going to make good progeny over cycles of breeding, we're always trying to predict breeding value. And just based on phenotypes, when we collect phenotypic data, we can predict breeding value just based on the phenotypes we observe. Because there is a component, that is of that phenotype that is due to breeding value. Okay, so this equation is just regression. And this is here, this is breeding value. that we're trying to predict. And for one individual, for individual I. Individual I. This one is the average breeding value of the population. And that's just a constant. So that's going to be the same across all individuals. And then this is the phenotypic observation. For individual I. And then this is the average phenotype. Of the population. And then this one, this is very important. This is a regression coefficient that tells us how well does the phenotypic observation predict the breeding value, or how do we translate the phenotypic observation to breeding value. So it's kind of helping us convert our phenotypic observations to breeding values. Okay. So basically, if we know we have a phenotypic observation and we know what this regression coefficient is, we can predict breeding value based on that phenotypic observation. And we'll talk, more about this coefficient and like what it means, like how do we get that coefficient. Okay. Well, what's next here? Okay, so there's another term I want to introduce, which is just transmitting ability. And it's just kind of an extra term that is pretty similar to breeding value, but it's the average effect of a random sample of half an individual's alleles. It's just half of the individual's breeding value. So if you take an individual and you randomly sample alleles, because that's what gets passed on, what's that average effect of all those alleles? And that's just half breeding value divided by 2. It's just taking, instead of all your alleles, it's a random sample of half of them. So it's just half of the total breeding value. That's transmitting ability. So if you look at what is, how do we go from there to predicting breeding value of progeny in a progeny population? So if you say, okay, each parent has a certain transmitting ability. It's got its breeding value, and then it transmits half, so its transmitting ability is half of the breeding value. So you have 2 parents, their breeding values. You can calculate what's going to be the expected average breeding value in the progeny. And it's just going to be the sum of the transmitting ability of the 2 parents. So that's here. So you've got, this is, this term, this is transmitting ability, parent 1. And this is transmitting ability, parent 2. Okay, now the AP, you know, this AP1, this is breeding value parent 1. And this one is breeding value parent 2. And this is the average breeding value of the progeny. So in simpler terms, it's just the average of the parents' breeding values. So 2 parents, you have their breeding values, take the average. Average of the parent breeding values is going to be the average of the progeny breeding values. Now, of course, there's variation in the progeny. So some progeny, that although the average is going to be the average of the 2 parents' breeding values, some progeny are going to have higher breeding values than the parents, and some progeny are going to have lower breeding values than the parents. So that's what we want. You know, we want to have, if the progeny are average of the parents, and that means we get something better than the parents, because you've got all that stuff that's above average is going to be better than the parents. So that's going to be really helpful to get something, you know, we're going to really be able to get something with a good breeding value, you select good parents, and then all the progeny that are above average are, you know, going to be better than what we started with. Okay, and so then we call that variation that we see in the progeny, we call that variation due to Mendelian sampling. So Mendelian sampling, all that really means is that when, so when 2 parents transmit their alleles, they're transmitting a random sample. So there is that, because each parent only transmits half. So it has to be a random sample. So by chance, it can be a really, you know, a lot of favorable alleles, or it could be a lot of unfavorable alleles. So there's, on average, it's going to be, you know, there's going to be an average, which is just that, if you repeated it, you know, infinite number of times, there's going to be this average, which is the transmitting ability. But then there's a variation just due to this random sampling. We call that Mendelian sampling. So then in a breeding value of a progeny, any one individual progeny is here. So that's average breeding value of the parents, which is this part. And then this is Mendelian sampling term. That just tells you how much better or worse is that individual from average. So you've got your population. This is the progeny. Let's say this is like parent 1, or this is parent 2. This is P1. This is like average. And then you've got an individual over here. So let's say this is individual AI, some progeny. This deviation from average is this due to this Mendelian sampling term. Yeah. Okay. Okay, so it's just Mendelian sampling term. Again, it's just how much an individual's breeding value deviates from average. And then it allows us to identify individuals that are superior to their parents. Also, we can identify things that are worse, but what we really want to do is identify things that are superior, better than the parents. Okay. Okay. I think I might stop for a break there. Let's do like a 10-minute break. Okay. Pause. Start again. Okay. All right, so we can start up again. And we were just talking about breeding value, and from breeding value, average breeding of a value of parents is average breeding value of progeny. And then we start talking about how progeny, there's a population and you've got an average, and then you've got individuals above and below average. And that kind of, we start talking about now populations and distributions. And so there's just some terms that we should go over before we get too far. We always kind of assume everything is normally distributed. And the mean, just the measure of the center of that distribution. The variance is the measure of the spread. And standard deviation is just the square root of the variance. Now, the nice thing about standard deviation is it has the same units as the trait. So that's convenient. This is just the definition of a variance, just mean of the squared deviations from the mean. I kind of like to visualize it. If you say this is your average and you have observations, individuals, let's say their phenotypes or breeding values, and they're above and, either above or below average. There's some deviations from the average for all these observations. Sorry. And take the squared, take these deviations and square them and take the average. So that's telling you what's kind of the magnitude of that deviation from the mean. And then just like we talked about how phenotype can be decomposed into different components, we can look at phenotypic variance and decompose it into different components. So we can say there's genetic variance and there's error variance. So these would be so if we take phenotypic values of individuals and we say for every phenotypic observation that we have, there is a proportion of that due to error and a portion of that due to genetics. So we just take those genetic components and determine what's the variance. And the same thing with the error. Those little error components, what's the variance? That's error variance. Then genetic variance can be decomposed even further to additive genetic variance, non-additive genetic variance. And so just by taking those individual, components across an entire population, we can determine different variances of all these different components of phenotypic values. So we said genetic variance, there's different components of it. So additive genetic variance, that's just the variance of the breeding values. So we look at it this way. Let's say this is average. Let's say breeding value. Or genetic, also genetic value. Sorry. So we've got, this is average, and then we've got, let's say red are some genetic values. And blue are breeding values. So let's say blue. So we've got genetic values, we've got breeding values. The variance of the breeding values, let's say these are blue. The sum of the squared deviations from the mean of these blue values are breeding values. That's additive genetic variance. And then covariance measures how two variables change together. So we take, just like in this example, we have two variables here. We've got breeding value, genetic value. So we look at the deviations between the mean and these points. So for the first variable, deviations between that variable and the mean, and the deviations between the other variable and the mean, and multiply and take the sum and divide by the total number of observations. So it's just telling us how two variables change together. How much do those deviations from the mean correspond to each other? And then correlation is just a normalized version of covariance. So this ranges between -1 and 1 and tells us the strength of a linear relationship between two variables. So it's just taking covariance, and here we're just dividing by standard deviation of x times the standard deviation of y. So our two variables are x and y. So it's just taking covariance and normalizing it so it ranges between -1 and 1. And so there's an important relationship that we'll use later, and that's the relationship between correlation and a regression coefficient. So this is just coming from some standard regression theory. So we've got, this is a regression coefficient for the regression of y on x. That's here. That's equal to the covariance between x and y divided by the variance of x. That's just a definition. That's also equal to the correlation coefficient between x and y times the standard deviation of y divided by standard deviation of x. So these are just, we were going to revisit these, but this just tells us how regression coefficients and correlation coefficients relate to each other, and covariances and variances. So all these things we can equate or solve for these different variables here. So this is going to be important later when we start getting into definitions or looking at how do we predict gain from selection, even heritability and selection accuracy. All these relationships kind of come into play there. Okay. So heritability, it's the regression of the breeding value on the phenotypic value. Okay, so in this case, we're looking at, so we've got the parent's phenotype and the offspring's phenotype. Okay, so the parents, so the parents' phenotype doesn't get completely passed on to the progeny. So only the breeding value gets passed on to the progeny. So how much those phenotypes from parent to progeny correspond with each other tells us how heritable the trait is. So we regress the breeding value on the phenotypic value. So let's say this, a parent's offspring is exactly telling us the breeding value of the parent, right? It's so that's kind of what we have on the y-axis. So you can think of this as breeding value. And this would be phenotypic value. So how much the phenotype corresponds to the breeding value. We regress breeding value on phenotypic value. The regression coefficient on that regression is equal to the heritability. So that's just a definition. The slope of the line from individual's phenotype to its breeding value. In this case, we're using an estimate of breeding value from the progeny. Okay. So that the progeny value is telling us the breeding value of the parents. That's why we have offspring phenotype here. Okay, so this heritability is a regression coefficient. So this and in this example, this is showing you here what does it look like if the heritability is 0.5. So you've got this relationship between parent and offspring, or parent and breeding value. And then if heritability is 0, there's no relationship. It's just a flat line. So the parent's phenotype cannot predict the offspring's phenotype. Or the phenotype does not tell us anything about the breeding value. Then heritability is 0. So the more that the phenotype tells us about breeding value, that's higher heritability. So if our heritability is 1, that means what we observe, the phenotype that we observe, 100% gets inherited. Like it will be, the progeny will look just like the parents, exactly, every time. If it's, and the closer it is to 0, the less, that our phenotype informs us about the progeny. Okay. All right, so since heritability is a regression coefficient, we can look back, what is that? What's the definition of that? How do we express that in terms of different variance components? So if heritability is a regression, right? We said it's a regression of breeding value on phenotypic value. That means based on what's the definition of a regression coefficient, we can express heritability in different ways. So we can say it's the covariance between breeding value and phenotype divided by the variance of the phenotype, which is also equal to additive genetic variance divided by total phenotypic variance. So you're probably thinking, this part is, how did we get from here to here? So this is a little bit hazy, right? So this is, again, this is the covariance between breeding value and a phenotype. Now let's look at what's, why are these the same? Like, why is covariance between breeding value and phenotype equal to additive genetic variance? And this is why. So if you say covariance between breeding value and phenotype, we can break this down. We can say there's a covariance between breeding value, and then this is phenotype here, which is breeding value plus error. So we're just saying, we're just spelling it out. We're saying y, the phenotype, is equal to breeding value plus error. So it's covariance between breeding value and breeding value plus error. Okay, so then we can express it this way. We can say, okay, based on properties of covariances. So this here, again, we can break it up and say this is equal to covariance of breeding value in itself plus the covariance between breeding value and error. Now this, what's the covariance between error and a breeding value? It has to be 0. There's no, because error is completely random. It's not associated with breeding value. It's just residual. And so this has to be 0. Right, so now what we're left with is the covariance between a breeding value and a phenotype is the same thing as additive genetic variance. It's not intuitive. Don't try to think about it that way. Just look at the formulas. It's not that intuitive to me anyways. So this covariance between breeding value and phenotype is equal to additive genetic variance. So that's how we end up with the formula for heritability being additive genetic variance divided by total phenotypic variance. It's because, what heritability is, it's the regression of parent to offspring, parent to breeding value, or sorry, individual and its breeding value. It's a regression coefficient. What's the definition of a regression coefficient? It's covariance between breeding value and phenotype divided by phenotypic variance. And that numerator is just the additive genetic variance because there's no residual covariance. There's no covariance between residual and breeding value. So, yes. Yes. Yeah. And I argue like heritability, like for me, narrow sense is the only thing that makes any sense. Because we're thinking about it. Think about this: parent, offspring, heritability, like heritability. Just think about inheritance and inheritability. Like how much does it inherit from the parents? So yeah, it's narrow sense. And we'll talk more about like other definitions of heritability, but for me, broad sense heritability doesn't make any sense. It's like, because it's including components that don't get inherited. So it's kind of like the term, I don't know, maybe we should come up with a better name. I think somebody else probably come up with a lot of different names, but we should probably stop coming up with different names for things actually. So, all right. So now that should make sense. We've got phenotypic value, breeding value, regression coefficient of breeding value on phenotypic value is the heritability. And we can say, okay, it's a regression coefficient. The regression coefficient is equal to this, the ratio of these two variances, additive genetic variance over total phenotypic variance. Okay, so there's another important term, which is the square root of the heritability. And that's important because that's actually the accuracy of selection based on phenotypic values. And we'll see that like when we get into the breeder's equation, that variable fits right in. So it's a correlation coefficient between a breeding value and a phenotypic value. Pretty similar to what we just talked about here, but now instead of a regression coefficient, we're talking about a correlation coefficient. Sorry. So yeah, what's the correlation between our phenotype and our breeding value? And that tells us, basically, this is our predictive ability of our phenotype. How well does our phenotype predict breeding value? So just looking at the definition again, what is a correlation coefficient? The covariance between X and Y divided by standard deviation of X times standard deviation of Y. Okay, I think I didn't define it further. It turns out, so yeah, this turns out to be H. I didn't go through the algebra, but we can look at those properties of variances and covariances again. And it turns out to be this additive genetic standard deviation divided by the standard deviation of Y. And sorry, this X, you can think here, you can substitute X for A for breeding value. This is just generic X and Y. Okay, so then heritability is always with reference to a specific population. So in like the strict, most purest definition of heritability, it's the proportion of the total phenotypic variance due to additive genetic factors in the base population. So this is in a population that has not yet been under selection. It's just a Hardy-Weinberg equilibrium, not under selection. It's this base population. It also has not been inbred. It's non-inbred individuals. And so that's like the strict definition of what heritability is. Anything else, or anything else that you measure, you can call it heritability, but this is like the most, I guess, strict definition of it. I'm trying to give you an example. So, let's see. Yeah, I think that's okay. So yeah, there's, as somebody just asked, there's a lot of different kinds of heritability, or a lot of different, it can mean different things to different people. So there's broad sense heritability. And in that case, the non-additive variance is included. So that means it's telling us, it's not necessarily telling us how much that phenotype can get past progeny. It's kind of telling us how much does that phenotype, how reproducible is that phenotype, basically. That's really what it's telling us. And a lot of times we'll just use a broad sense heritability estimate and assume that it's pretty close to narrow sense. Like there isn't going to be much difference, because if we think about the infinitesimal model and our assumptions about additivity versus non-additivity, we assume that additivity explains a lot of the variation. So there shouldn't be a lot of difference between a broad sense and a narrow sense heritability, if we can make that assumption. Okay, and then you might have heard of this term, line mean heritability. This is also a broad sense heritability, but it's looking at variance among lines, genetic variance among lines, overall, divided by total variance, total phenotypic variance. The difference, I guess the distinction there is, I can draw. So I said something about how heritability, in its basic definition, it's this additive genetic variance over phenotypic variance. And these variances are, so, and yeah, this is with reference to a base population. So a population, so this, especially this additive genetic variance here, is from, sorry, within a non-inbred base population. Okay. So if you've got, let's say you've got a population where individuals are slightly inbred. Inbreeding increases that additive genetic variance. So among individuals, that variance is going to be larger. So that's why we're always talking about, if in this, pure definition of heritability, we're talking with, it's got to be with respect to a base population that is not inbred. So this is, yeah. Okay. So if we've got, let's say we want to look at heritability, but line mean heritability. So now we're not talking about non-inbred individuals. So we're talking about, so this is like line mean H2. So now it's going to be now this variance is genetic variance among lines. Which is going to be, if they're completely homozygous, it's going to be 2 times the additive genetic variance. Okay. This is probably more common actually than, I think, in depending on the context. I think in plant breeding, we're mostly talking about this line mean heritability, because we're usually interested in some kind of inbred individual at some point. So this is usually what we're talking about. But keep in mind that there are different kinds of heritability. And so if you, and there are different, additive genetic variances within, is going to be different within a base population that is not inbred than among lines that are inbred. So it's going to depend largely on the level of inbreeding. Okay. So then, actually, I forgot to mention, so when we're talking about a base population, if you want to try to envision a base population, these are going to be like individual plants, like non-inbred plants. And, so obviously not very realistic to work with in like a real population. We're typically not going to measure phenotypes on like single plants and try to estimate a heritability. So it's kind of a good theoretical concept, but kind of in practical use, this is what we're using a lot of the time. Okay. Let's see what we got here. Some questions. If we estimate narrow sense and broad sense on inbred lines of a self-pollinated crop species, do they converge? I mean, like, are they similar? Is that the question? Um, I would say yes. I don't know. I don't know. Yeah. Okay. Yeah. Okay. I think that's for now. Okay, so I think this will be our last set of slides before we take a break for lunch. So this is kind of the last of the really basic like theory. And then after lunch, we can talk more about genomic selection specifically. Okay. So I want to talk a lot about response to selection and how do we predict response to selection, because that's really important for the rationale, like why we even want to do genomic selection in the first place, and it'll help us understand how to apply it and what's the point. And so we talked, this whole session is just about response to selection, and we're kind of lucky to be here in Illinois, where we have the world's longest running selection experiment in corn. So this is from 1896. This graph is showing results from 1896 to 2016. And this is where, selection was done for low protein, high oil, low oil, and then a lot of different selection treatments were imposed at different points in time, looking at how long, can we, so can we select in one direction, and then can we reverse, so, select in the opposite direction? Do we still have variability? So there was a lot of interesting results. But I think one of the big lessons learned from this experiment was that you can achieve gain for a really long period of time without just within within one population, without bringing in other material, other parents, without any other variability, just from that standing variability that existed in the original population. And this is another interesting graph of the same experiment where we've got oil content. And so there's, in the low direction, we actually reach a point, there's kind of a limit, because there needs to be a minimum amount of oil in a seed, for it to be viable. So we do really reach kind of a selection limit in that direction, but in the high direction, we're not really reaching a plateau. Somebody once pointed out this, like, why this variability, and it turns out this has nothing to do with the genetics. This is actually just how the phenotype was measured over time. So they changed the phenotyping method. So now that's why you see more variation in later years. But the point is, we can still, we can achieve gain from selection over time, and we still maintain variability, which we can, further make gains. And then response to selection, it's just another way to say genetic gain. That's that average. It's a change in the population average from cycle to cycle, from one generation of breeding to another. So if we take, and we're talking about genetic values here. So if we look at this graph, so let's say our base population, where we have done no selection previously, is in dark blue. If we impose, one cycle of selection, so we take the best individuals, intermate those, create a new population, we generate this selected population, there's going to be some improvement because, we selected the best, we recombined, we generate a better population. In the next cycle, we take that, what we just generated, our most recent cycle of selection, and again, select parents, recombine, create the next selected population, and then we compare it to our base population. Now we have made more progress, and so on over multiple cycles. We continue to make some incremental progress every cycle. So that means, so this response to selection or the genetic gain, it's just the difference between the average genetic value in the population from one breeding cycle to the next. And we can predict selection response just based on these two principles. First is that the breeding value can be predicted from phenotypic value using this linear regression equation that we introduced earlier. Okay, so we remember we had this where we had breeding value of an individual can be predicted based on average breeding value of the population, plus this regression coefficient of breeding value on phenotype, and then the phenotypic value of the individual minus the population average phenotype. So this actually helps us, we'll see, I'll show you how it helps us predict response to selection. So we can take this and rearrange it. So now all I did was move this average breeding value to the other side. So now we have breeding value minus population average equals this regression coefficient, which is then multiplied by the phenotypic value minus population average. Okay, so then this, remember this regression coefficient is, or this is just a visualization of this formula here. So you've got phenotypic value is on the X, breeding value is on the Y. What relates these two is this slope, the regression coefficient BXA. So you've got this is the slope. This is over here. This is on the X-axis, this is on the Y. This is what we're trying to predict. The breeding value of the individual minus population average. So if we know this regression coefficient and we know the phenotype, we can predict the breeding value. Okay, so does anyone remember what the slope is from, like, I think the last lecture? If we're talking about phenotype, like phenotype value and breeding value. This, who said it? Oh, heritability. Okay, yeah. So this turns out to be heritability. So then now it becomes more familiar. So it's like we know the phenotypic value, we know the heritability, we can predict the breeding value. Okay, so now we're just talking about a single individual's breeding value, but the same can be applied to groups of selected individuals. And that's important because when we're doing selection, we're selecting several individuals to intermate them. So we can, the same exact formula, just say now instead of just AI and XI, we have A star and X star. And this is referring to a group of selected individuals. So this group of selected individuals, breeding value, average breeding value, minus the population average. And here on the X side, this group of individuals, selected individuals, phenotypes minus population average. So this is just the same thing, but applied to multiple individuals now. Okay, so that's the basic principle number one that is going to help us predict breeding value, or sorry, predict gain from selection. And then the second basic principle, which we've already talked about, is that the average true breeding value, or predicted breeding value actually, of two parents is equal to the average breeding value of the progeny. So here you've got breeding value of parent 1 here, breeding value of parent 2. So the average of the 2 parent breeding values tells us the average progeny breeding value. And the same can be done actually if we have predicted breeding values, the same relationship holds. Okay, so this is just illustrating, again, if you have 2 parents, the average breeding value of the progeny is just going to be the average breeding value of the parents. Okay, so now we're going to make the connection. How do we use those 2 principles? So that regression equation plus the prediction of breeding value between parent and progeny. How do we use that to predict the selection response over cycles? Okay, so we start out with this. Let's say this is our population 0, where we have done no selection previously. We're just starting out. And we take these individuals. This is A star. And this is a distribution of breeding values. Okay, so we take, this is A star. Okay, and we know that, this average of these individuals minus the population average, that's this part right here. So that's what this part is. We can predict that using the phenotypic value and the regression coefficient between phenotype and breeding value. We also know that based on the second principle, that breeding value of parents is average breeding value of progeny. So now we can say that all these individuals that we just selected, if we intermate them, the breeding values of the progeny are going to follow this distribution. So they're going to have the same average as A star. Okay, because average breeding value of the parents is same average breeding value of the progeny. Okay. Okay, so now let's look at this. We've got breeding value. Now remember what response to selection is. So we've got breeding values of population 0 and breeding values of population 1. So we've got the average of that population 1 minus average of population 0 is the response, just by definition of what is response to selection. It's average of population 1 breeding values minus population 0. So that's the response. And remember, this is A star again. A star is our average of our breeding values of our selected individuals. Okay, so we know R is actually A star minus A bar, which is population 0 average. Okay, so this is going to be response. So now we can actually start substituting. So we can put this R here because R is equal to this A star minus A bar. So it's this breeding values of the selected, average breeding value of the selected minus average breeding value of the whole population. So we can just substitute it. Okay, so we plug that in, and then what we get is response is equal to this regression coefficient, BXA, and then multiplied by the phenotype, average phenotype of the selected minus population average. Right? So based on this, so this is the original equation based on principle 1. Right, based on the second principle, we get this one. So this response is just equal to A star minus A bar. Now this next part, this part where we have the phenotypic values, we usually refer to this as the selection differential. this is the phenotypic. So if these are the selected individuals, here, average phenotype of the selected minus population average phenotype. We refer to this as selection differential. So phenotype average of the selected minus population average phenotype. So now we can just substitute this with this new variable S, selection differential. Okay, so that's what we do. And remember this regression coefficient, BXA, regression of breeding value on a phenotype, is just heritability, the H squared. So then what we end up with is this R equals H2S for just phenotypic mass selection. Now we can keep going and keep generalizing it. This selection differential, right now it's in units of phenotype. So we actually have to know, we have to actually have the population, measure its phenotypes, you know, have our selected individuals phenotypes and have the data, right? But we don't have to, you know, we can actually express it in standard deviation units. And then, you know, we can generalize the formula a little bit more. So let's say we want to express S in units of standard deviation. So we want to say, we don't want to say like, we don't want it to be in units of the trait, like, you know, grams or inches. We want it to be in standard deviations from the mean. So if we take S and divide by the standard deviation, then we get this value K, which is just the standardized selection differential. It's saying how much, so now the individuals that we select, how much are their phenotypes better than the mean in units of standard deviation? Are they 2 standard deviations away from the mean, 3, you know, in units of standard deviation? So that's going to be K. So K is just selection differential divided by standard deviation of X. If we rearrange it, S, just by algebra, S is equal to this K times the standard deviation, the phenotype. So now we can plug this in for S. So we have this, right? We can plug it in. And this is what we end up with. Response is equal to this regression coefficient of, you know, to predict breeding value based on phenotypes. K is the selection, well, we refer to this as the selection intensity. And this standard deviation of X is just the phenotypic standard deviation. So basically tells us kind of how much away from the mean in terms of in units of standard deviation. And then we need to, if we want to, you know, express our response in terms of selection intensity, we have to add this variable standard deviation of the phenotype. Okay. All right, so now we have this, which is already pretty generalized, but we still have this variable, which is this regression coefficient, which is, if we're just focused on phenotypic selection, that's easy. We can just substitute that for the heritability. But if we're using a different type of selection, let's say we're not just doing phenotypic mass selection, and let's say we're using a genomic selection model. So now it's actually better if we can replace this with a different, with something else. We want to replace it with selection accuracy. Okay, so selection accuracy. If we want to express this in terms of selection accuracy, we have to go back to this relationship between, you know, what is selection accuracy? Correlation between breeding value and phenotype, which is then covariance between breeding value and phenotype divided by the variance of the breeding value and the variance multiplied by variance of the phenotype. So we can just just spell it out. How is this defined? Do some rearranging. Okay, so we again look at what's the definition of a regression coefficient. Okay, so it's this covariance between phenotype and breeding value divided by the phenotypic variance. All right, we can, so this is, so I just took that and the denominator, sorry. What am I doing here? So just look at the denominator. So I took the the phenotypic variance, broke it down into then phenotypic standard deviation times standard deviation. Okay, now these are starting to look, we can start to see some things in common here. So we can look at this is starting to look pretty similar. So now we can see, okay, this regression coefficient looks pretty similar to the correlation coefficient. So if we just multiply, let's see, if we just multiply RXA times additive genetic variance. So let's say we multiply this. Sorry. If we just multiply here, this is just algebra. These will cancel out and we'll end up with this. RXA, so this correlation coefficient between breeding value and phenotype multiplied by additive genetic variance divided by the phenotypic standard deviation is equal to this regression coefficient. So now we can plug, remember we wanted to plug this in. We wanted to eliminate regression coefficient from the formula. So now we have this part, which we can substitute here. So now we can put, we don't have any regression coefficient, we just have correlation coefficient, which is our, prediction accuracy, you could say, or predictive, prediction accuracy, selection accuracy for the correlation between our, phenotype or breeding value, or if we're using an estimated breeding value from genomic selection, it will be the correlation between our estimated breeding value and the true breeding value. Okay, so we can plug this in. Okay, then there's some things that cancel out. So this already cancels. Yeah, did that already. And now we get this formula. So it's basically, so it's just, this is response to selection, from each cycle of selection. This is the correlation between what we're using for selection, whether it be a phenotype or an estimated breeding value, and the true breeding value. This is additive genetic variance, and this is the selection intensity. So now this is more generalized. So it's, we can use this for phenotypic selection, we can use it for genomic selection, a lot of different, and we can rearrange, we can go back and rearrange this formula, this, start back from the beginning and rearrange in a lot of different ways. So you'll probably see, you can see this response to selection formula. There's a lot of different versions that are equivalent. It's just how the algebra was done to, rearrange it. So there's a lot of different ways. But basically, it's just those two principles about parent breeding value. The average of the two parents' breeding value is the average of the progeny breeding value. That's the main, that's basic principle one. Basic principle two is that you can predict breeding value based on phenotypic value using that regression equation. So those are basically, the two main principles. After that, it's just a bunch of algebra rearranging, and then you get this formula. Okay, Breeder's equation. So I think we've got, yeah, we've got about 15 minutes until lunch. So I think we should definitely finish this lesson before lunch. Just have a few more slides. Okay, so the Breeder's equation. Response per breeding cycle. Yep, that's what we're trying to predict. Selection accuracy. That's that correlation between whatever we're using for selection and breeding value. And then we've got additive genetic standard deviation. I might have said variance earlier. So yes, it's additive genetic standard deviation among, in this case, it's among the selection units. So it's among whatever we're selecting among. So if we're selecting on inbred lines, it's going to be additive genetic standard deviation among those lines. If it's single plants, it's going to be among those plants. And then we have K, which is the selection intensity, which is just that how far away is our selected individuals from the population mean for whatever we're observing, whatever we're selecting, whether that be phenotypes or breeding values. Sorry, estimated breeding values. So it's whatever that is in units of standard deviation. Now, what's nice about this using selection intensity is we can actually pretty easily obtain this value just by knowing the percentage that we're selecting. So we have our distribution. We have this group of individuals that we're selecting among, and we select a certain percentage, P percent. By properties of the normal distribution, we can determine what's going to be K. So say you take this, this is what we call the truncation point. So this, this distance between the x-axis and the, this is the x-axis and the curve and the normal distribution. This is what we call Z. So if you take, this height here is Z divided by the percentage selected, you get K. Or K is the average here. Sorry, it's the average of the selected minus the population average in standard deviation units. And then if you want to just calculate K in R, it's easy. You can use D norm Q norm P divided by P. So that's handy. There's usually, you've probably read it in books, there's like tables, you can look it up. But here you don't have to do, you can just calculate it. Let's say Q norm, D norm, Q norm P divided by P. So this part is going to be this is going to give you Z. All right. So now another really important concept is actually gain per unit of time. And it's not that complicated, but it's, pretty critical. So we're normally interested in not just gain per cycle, but gain per year, or, per other unit of time, usually year. So if we have, and the reason for that is if we can have a breeding cycle that is like 15 years long, and let's say response per cycle is good, but if you think of, you get that response over a 15-year interval, then the response per year is going to be that total response divided by 15. So then on a per year basis, the response may be small. So we want to increase the response that we get every year in the population. And if you look at just genetic trends, so this is percent oil, this is back from the original example of the selection experiment. So we have percent oil, and we've got the year. And every point on here is a population average. The slope of this line is actually the response per cycle divided by the L, the time required to complete one breeding cycle. That's the rate of genetic gain. So this is, the slope. Which is genetic gain, rate of genetic gain. Okay. So that's going to tell us how much does the population improve every year. And we want that's what we want to maximize actually. So, we can increase R easily by just like delaying, making the breeding cycle longer. Say you want to increase accuracy, let's say you want to test your lines for like 10 years. And you say, I have the most accurate selection in the world. Now I'm ready to make crosses. So you would like maximize your response per breeding cycle, but because you just made the breeding cycle really, really long, now you have to remember, like, this is divided up by how long it takes you to complete a cycle. So yeah, you want to that's what you want to maximize. You want to get the slope higher. You want to increase the genetic gain on a per year basis. Okay, so before we wrap up, first, there's a lot of assumptions with the breeder's equation. So it's not going to be perfectly transferable or accurate in every case. So one of the assumptions is that you've got discrete generations, which means that with every new cycle of breeding, you don't use previous breeding candidates. Like, you generate that new population and then you only select individuals within that new population. You do not select previous generations. So that's an assumption that's usually not all that realistic. Then the other assumption is that there's a constant variance. So that means with time, we're not actually changing the genetic variance. It's remaining constant. Also, accuracy is constant, selection intensity is constant. All those things we're thinking about predicting rate of gain over time. Those variables that we're plugging in, we're assuming that those are not changing. And that constant variance, it tends to be relatively realistic if we can say that our traits are following that infinitesimal model of inheritance. Because remember, with the infinitesimal model, we said that selection does not affect the variance. Because there's so many small loci, like you're not really changing individual loci frequencies. Okay, and then the third one, truncation selection, just means that we're selecting that upper tail of individuals. We're not selecting, everything that is below that truncation point is just getting discarded. So these assumptions, although, we tend to kind of violate this, basically, you can think of it as the breeder's equation gives you a nice framework, to look at variables that affect gain from selection. So kind of gives you some guiding principles, but kind of in a very complex breeding situation, you're not going to be able to just like plug numbers into that formula and go. Like, it's not gonna be that accurate, because you've got a lot of assumptions that are being violated. And so although it's a good framework to follow, it's not going to give you some like precise results on what is going to be your gain from selection. So if you really want to get that kind of information, there are ways, there are different additional formulas that you can use. But if you're really wanting to get a very accurate result, you'll have to do some other kind of simulations, simulate the whole program, every step, and in order to be able to get away from making these assumptions. Okay, so there's some key points. We can derive the breeder's equation from those two principles that I mentioned about breeding value of the parents and breeding value of the progeny being the average of parents equal to average of the progeny. And then that there's a regression relationship between phenotype and breeding value. And when we define the equation, we can clearly see that gain from selection per unit time depends on selection accuracy, additive genetic standard deviation, selection intensity, and length of the breeding cycle. And there are some assumptions in there that we sometimes violate. So if we really want to precisely predict gain from selection in a breeding program, we probably have to do some other simulations. We will skip that, but if you want to take that during lunch, it's up to you. So I'll leave that there. But yeah, this is a good stopping point if you guys have questions. Let's see. Okay, so there's a lot of questions about the breeding cycle. So what is that? So it's from the time that you make a cross to the time that progeny from those crosses are then used again as parents. Yeah, so it's, as Valerio said, it's the cycle is from the moment advanced progenies become parents again. It's a good way to put it. So somebody said, okay, so from time of crossing until the time of launching a new. That's actually not necessarily the case. Like, you can select parents before you release it as a variety. So you can say, you make cross, you generate the lines, you start testing, maybe you have 1 or 2 years of data, and you decide, I'm going to use it as a parent. That's the end of your breeding cycle. Now, you may actually need more time to test to release it as a cultivar. So so that I would say that it kind of depends on the breeding program. Some breeding programs would probably not make a cross until it's been released as variety, and then it will take like 10 years. Okay. Somebody's asking about inbred, because inbred lines don't have heterozygous genotypes, so that's why it's more due to additive effects. So I think what this is referring to is, in inbred lines, you don't have dominance because you don't have heterozygous genotypes. So there's going to be no dominant genetic variance, but you can still have interaction variance, and you can still have, error variance. So you do eliminate the dominance, but there can still be other non-additive effects. And the message, recommended textbooks. Yeah, I can send you some of those. Okay. Maybe we'll stop there. Take a break, so then we can come back by 1, hour and a half.
I need to go.
I forgot about that. We have to water. Okay. All right, we can start back again. So this afternoon, we're just focusing on genomic selection. First, we'll just give an overview, how it works. And then I'll talk about factors that affect response to selection in genomic selection, and then we'll do the exercise. Okay, so this part, we're going to be talking about, what's genomic selection, how's it's applied to breeding, and then kind of basic guidelines for implementing genomic selection. We're going to talk a little bit about genomic bluff and ridge regression bluff and understand those models a little bit. So genomic selection is just the use of genome-wide marker data to help predict breeding value for the purpose of selection. So it's just anytime we're using a model that includes marker data to help predict breeding value. And then the key part of obviously is we're using that for selection. It's useful for traits that are conferred by at least 3 loci. So remember, we are talking about how quantitative trait variation is brought about by multiple loci. Genomic selection can predict that quantitative variation in breeding value. If we have fewer than 3 loci, probably we don't need a genomic selection strategy. Probably just having single markers and selecting based on single markers is enough. But if it's more than 3 or 4 or 5, even, if it's getting to those larger numbers, then, it becomes more difficult to predict based on individual marker genotypes. Imagine if you have in front of you, let's say you have a trait that's affected by 5 different loci, and you get some marker genotypic data back, then it's starting to become difficult to tease, to say, how do I select based on these 5 independent marker loci genotypes? So then in that case, you're probably better off using some kind of a prediction model. And there are genomic selection models that can handle those kind of traits that are based on smaller numbers of markers and traits that are, follow more of an infinitesimal model. So it's not necessarily need to be the case that the trait is affected by thousands of markers. It can be even for more simple traits. And then I think another important thing about genomic selection is it can be pretty disruptive to a breeding program. Like what it allows a breeding program to do can, potentially totally change how the breeding program is structured. And, how the kind of data that you need to generate to train an accurate prediction model also may require you to change some of the things in the selection process. So say, for instance, if you have a breeding program and you're just culling, like visual selection, you're culling out a lot of genotypes. If you're not actually collecting data on that, you're not going to be able to predict it later. So those are sort of the things that may be different in a strictly phenotypic selection based program. There are some things you can do, but that would not necessarily help you in the genomic prediction context. Like you're not going to be able to train a model if you do all that culling, so that's just an example. So there are different things that you might have to change. And then what's the purpose? Like, why do genomic selection at all. So there's kind of two different things that it can let you do. And in some cases, depending, and it all depends how it's applied. In some cases, it can help you increase selection accuracy. And in some cases, it can help you reduce the breeding cycle time. With reducing the breeding cycle time, the basic idea is you can genotype and predict your breeding candidates before you actually can phenotype them. So as soon as you have your candidates, whether they be, they can be lines, it can be individual plants, you can genotype them, you can predict them and select them as parents. That's the key part, what allows you to shorten the breeding cycle time, is that time to parent selection. And that's actually the application of genomic selection that has the most potential to accelerate the rate of gain, is being able to select among your candidates before those candidates are even tested. But, that's, in some cases, we can just apply genomic selection to increase accuracy. And in that case, what we're doing is we're taking phenotypic data on your candidates. And in addition to that, using the marker data and using historical data on relatives that have been phenotyped and genotyped. So in that sense, genomic selection can just be kind of a way to add more information to your selection. So there's this kind of two different applications. So if we look at just the increasing selection accuracy side of things. So if you have a breeding strategy like this, let's say this is kind of a typical self-pollinated crop strategy. So you cross parents, and then you have some F1s, self-pollinate them a few generations. And once you get to a point, let's say you develop a line and you genotype it, and you can phenotype it as well. So because you've already generated a line, you can generate phenotypic data on that. In addition to that, also genotype it. So you have both phenotype and genotype together, and you're using all that information to predict breeding value. You can also use data from the past, related lines that have been phenotyped and genotyped. Then you can make those selection decisions, say for advancement. Then you can, again, in the next cycle, in the next stage of testing, phenotype and predict breeding value. And then at some point, you select parents, to the point where, say, you feel like you have enough accuracy to select parents. So that's, in that case, it's really not much different from any typical breeding pipeline, just generating lines, phenotyping. The only difference is now we're just adding that extra data and being able to use data from the past on relatives and just using more information. And you can see, like in this case, the breeding cycle, I just put it as 4 years. Let's say the time that it takes from crossing the parents to the time that you will start selecting parents out of those progeny. So that's like, I just put it, suppose it's 4 years. That'd be the breeding cycle. Now, if you want to reduce the breeding cycle time, let's say we have the same breeding strategy now, we're gonna use GS to reduce breeding cycle time. So we do the same thing, cross generate let's say generate a line. And now instead of waiting a few years before we select parents, like in this case, we selected parents after evaluating phenotypically a couple of years. So in this case, we'll actually just genotype those F5s, predict breeding value, and then select the parents again. So we're closing the loop sooner. Suppose now our breeding cycle time is 3 years. So we're just, we reduce it by 1 year because now instead of having that extra year of testing, we're selecting parents right away. And in this case, our selection accuracy is probably going to be lower than what it would be if we used phenotypic data. So if we actually took those F5s and phenotyped them first, well, we should have a higher accuracy than if we were to just genotype them and predict them. So that can be a little bit confusing because remember I said here we can actually increase, like with genomic selection, we can increase accuracy. And on this side, I'm saying we reduce accuracy. So just to clarify that a little bit, if we're using, the best accuracy is going to be both phenotype and genotype together. So on your candidates, you actually observe, you phenotypically observe your candidates and you genotype them and you use data on their relatives to help improve accuracy. So that's going to be the most accurate because you just use, it's the most information, using more information. Then the next most accurate is going to be phenotypically observing your candidates, actually phenotyping them. That would be the next best thing in terms of accuracy. And then the lower accuracy is going to be when you just predict it and the candidates have not been phenotyped at all. So you've never seen those candidates at all, just phenotype them and you're just predicting them. That's going to have lower accuracy. And that's where the reason why that can work, that can actually accelerate gain, is by reducing the cycle time. So even though the accuracy may be less in this scheme where we're selecting before phenotyping, because we're reducing the breeding cycle time, we can still get a faster rate of gain. So it all really depends on that the accuracy versus the time. So in this case, if in this scheme, let's say my selection accuracy is, suppose it's 0.5. So let's say and then your breeding cycle time is 4 years. Remember in the breeder's equation, we've got accuracy is in the numerator and time is in the denominator. So let's say we've got 0.5 and the time is 4. And we have some other variables here. We're not going to worry about those. In the other case, oops, sorry. So in the other case, let's say our accuracy is less. Let's say our accuracy is 0.4. And the time is 3. And we have some other variables in the numerator. We're not going to worry about those. So then what's going to be better, in terms of gain? In this case, we've got more accurate, but longer breeding cycle. In this case, it's less accurate, but a shorter breeding cycle. So the only way to really compare, which is going to be better is by really just solving it and figuring out, okay, does the accuracy that I lost offset, is it offset by the time that I've gained? So there's that balance. All right. So before I talk about this situation where we reduce the breeding cycle by just 1 year, we generate a line, then start doing our prediction and selecting parents. You could actually go more aggressive and before you actually generate a line, genotype individual plants and then select parents among those. And that will give you the biggest reduction in the breeding cycle time. So instead of reducing from 4 years to 3 years, you're only gaining a year. If you go from 4 years to half a year, that's like a huge difference. So now that's where you can start to see if you could get a reasonable accuracy in this scenario, you could accelerate gain quite a lot. So let's say if if you reduced your accuracy in half from the original strategy, you could still like I think times 4 your genetic gain, multiply times 4. So just because you can reduce the cycle time so much. At the same time, at the same time that you can select before generating lines, you can also you can also genotype lines and improve accuracy during the advancement stage. So in this case, you're increasing your rate of gain here in your by reducing the cycle time. And then you can also, if you want to, you can also genotype and predict breeding value at these later stages and improve those advancement decisions. So it's not, either or. And you still also, it's important, you still have to genotype these lines and phenotype them, because all this data is going to help you train the model that you're going to use to keep predicting every cycle. So you're still going to have to phenotype those lines and genotype them, and then use it to train the model. So in a sense, in this strategy, that's kind of the disadvantage, is you're having to genotype your candidates here, and you're having to genotype your advanced lines later. So in this other scenario, you're only genotyping advanced lines, then you're using them for candidates for parents, and then you're phenotyping them, using them to train the model. Now, in this case, you have candidates that you're genotyping, selecting parents for crossing, and then after you generate lines, you have to genotype those as well. And so there's like extra genotyping involved in this case. Okay, is anybody? Yes, people are asking questions, so I'm gonna just check. Okay, somebody asked that. What's the ideal stage for fixed line? In the polyploid crop. Yeah, I haven't really thought about genomic selection in a polyploid crop, but it doesn't necessarily, I mean, the examples that I'm giving are with self-pollination involved, but it doesn't necessarily have to be self-pollination. I mean, all you need is you need a way to make crosses, generate progeny, genotype those, and cross them again. That's, the key recombination part. And you need a way to train the model. So you need a way to recombine, genotype candidates, select, and then you need a way to generate some kind of clone, or generate something that you can phenotype, whatever that may be, a line, a clone, even if it's like, let's say you're working with trees, it'd be like an individual tree, or, whatever it is that you normally phenotype, generate that, phenotype it and genotype it, and use it to train the model. So those are kind of just the key parts. This is an example with self-pollinated crops. If you were working with hybrids, you would have to be generating some lines, then generating hybrid, and then using that hybrid information to train the model. So it's whatever you normally phenotype is what you're going to use to train the model. You're going to have to generate those entities, phenotype them, genotype them, train the model. And then what you cross, when you intermate, and what you select from, those can be just individual plants, or they can be what you would normally phenotype. It's pretty flexible. But in this most aggressive, strategy, you're working with individual plants. Okay. somebody's asking about, okay. How repeatable must the SNPs be? So is that question referring to, like, do you get the same markers every time, or, yeah. So, I mean, you just need some way to be able, so with your marker data, you need to have some way that there are markers in common between your training set, the data you generate for training, and what you're genotyping for your candidate, to select your candidates. So if your data sets, let's say you have completely different markers, like you genotyped some markers over here with this SNP platform on your training set, and now you have a new SNP platform on your candidates, you need to have some way to connect them so that there is like some common variables between those. So there's, if you have the position in the genome, you should be able to do imputation, especially if you've got individuals that have been genotyped with both marker sets. So in general, I don't think it's a big problem if there's markers not 100% in common, but it's just adding a lot more extra steps to, trying to get the data to combine. So someone's asking, should you choose SNP chips rather than GBS? I don't know. I mean, I think you want something that's, I think for your markers, you want it to be cheap and you want it to be quick. You want to be able to genotype heterozygotes, and what's the other criteria? You want it cheap, you want it quick, you want to generate heterozygotes, and you want to have some common markers between every time you genotype. You want to probably have some markers in common. And I think beyond that, then depending on the platform, I think if you just think about those criteria, then you can choose the platform. Because, sometimes SNP chips may be really expensive, or the turnaround time is slow, or GBS maybe turnaround time is slow. I don't know. It depends. I think every situation is kind of different. So maybe there will be a new platform that is better than all of those. That's what I hope. So. Okay. We'll talk more about marker density in a minute, so I'll get there. But I want to point out, though, that genomic selection decouples population improvement and variety development. In this, what we just talked about, where we were selecting our candidates at an early stage, like prior to generating lines, this is really the population improvement cycle. So this is where we're just crossing, selecting, crossing, selecting. We just continually cross and select. And the selections are done based on predictions, genomic selection. So that's the population improvement part. And then there's variety development, where from that population improvement cycle, some of those individuals get self-pollinated, or you generate clone, whatever it is relevant to your species. And then you evaluate those, and from there, release varieties. So, and also use that same data for training the model. So this is the variety development kind of cycle and the population improvement cycle. So these are now decoupled. So we don't need to wait to release a variety to select a parent. Everything is happening at a different pace. So this population improvement cycle can be much faster than the variety development cycle. And also they're kind of separate activities in a way. Okay. So then you might have heard people talking about, or even I've already mentioned it probably, the model training set. What that means, that's just the set of genotypes used in the genomic selection model. That these are genotypes that have phenotypes for the primary traits of interest. So this is any information that you are going to use to train the model that have to be genotyped, have to be phenotyped. And the model training set is also referred to as training, this is just in different people, authors have referred to it as training population, calibration set, reference set. There's a few, names, but these are all referring to the same thing. These are just sets of genotypes that you're going to use, and they have phenotypic data. And typically if you're working in a breeding program, this is just going to be whatever data you have from the past that you've generated in the testing program. So lines that you've been in testing, they're genotyped and phenotyped, you can use that to train the model. And the thing that is affecting accuracy the most is the training set. How big is the training set? What kind of lines, or what's the composition of the training set? How relevant is it to what you're trying to predict? That's a key factor that affects accuracy, much more than, number of markers or anything. So if you think about it, there's kind of, different options for how you apply genomic selection and train the model. So if you think about here, like in this yellow circle would be your breeding program germplasm, everything that you've generated, including your selection candidates, what you're trying to predict. And in some situations, your training set is kind of the same as your selection candidates. So maybe you've taken those candidates that you're going to select from, and you've also phenotyped them. So in that case, what you're trying to predict is also part of your training set. It's already has phenotypes. So there's sort of, you don't really have a separate training set from your candidates. Everything, if everything that you have is phenotype and genotype. So that's in one scenario. Now, in the other scenario, your training set does not include your candidates. So this is the case where, let's say you're trying to predict candidates that you've never phenotyped, then your training set's going to be other individuals that you have phenotyped. And there's no overlap between them. So everything that you're trying to, in this case, everything that you're trying to select has not been phenotyped, and you're using data from the past to try to train the model and predict breeding value. And then key thing to remember, everything, it should always fall within your breeding germplasm. So whatever your training set, your selection candidates, they're part of the same germplasm pool. They're part of that same big population. Okay, somebody's asking. Oh, someone's asking about what's better between these two. So it's sort of, this kind of relates back to maybe this slide here. So in this case where you're just going to increase selection accuracy and you're going to genotype and phenotype everything, they're going to consider it as candidates. That's where this scenario is relevant. That's where, you're not reducing the cycle time, but you're increasing accuracy. So that would be. In this case, you can have maybe lower accuracy, but then faster cycle time. So then what makes a good training set? And number one is the data should be relevant. So it should be from your target population of environments. So if you're breeding for a specific region, you should be testing a representative sample of that region when you're collecting phenotypic data. And then you should be collecting data on those primary traits that you're interested in. So make sure if your goal is to improve yield, you really should be measuring yield and using that to train the model. And then meaningful data. So that's meaningful is not the same thing as accurate. So accurate, there you can have you can have really accurate data for any kind of thing that you measure, but for something to be meaningful, it has to be actually predictive of what it would be in a farmer's field. So whatever you measure you have to have a confidence that it is going to be representing what it would really be in a production environment. So that's actually why we generate in self-pollinated crops, when we generate lines. That's a big reason why we do that. We generate fixed lines because you can't generate meaningful data on a mixture. So what we release as varieties are uniform variety, uniform genetically. So that's kind of the difference between what's meaningful versus accurate. Meaningful being relevant to the production environment. Accurate is just having less error, being more repeatable. So the third point is that the germplasm that you use for model training should be really closely related to your selection candidates. So the more closely related your individuals in your training set, the more closely related they are to the candidates, the higher accuracy you're going to have in your genomic selection model. So the most extreme case would be, let's say you take a biparental population and you generate F2s or recombinant inbred lines. Though individuals that are from that same cross are going to be pretty closely related. So if you went and took a percentage of that population and used it as a training set to predict the rest, your accuracy would be really, really good because you have all those individuals are pretty closely related. And there are some places that you actually use that as a strategy. So generate biparental populations, phenotype a subset, genotype the rest, and predict them. So that can be helpful. The only problem with that is the time cannot really be reduced because you're generating the family and then phenotyping a part of it. So you're not reducing breeding cycle time. You probably gain some other advantages, but time is not really saved there. Also, interestingly, about that strategy, if you use biparental populations, the training set size can be extremely small. Like I think you can just use like 50 individuals to train the model to predict the other individuals in the population. Okay, and then, just going off of that last point about how the training set has to be closely related to what you're trying to predict, you need to update the training set because every cycle of breeding, we're changing the population a little bit. So there's recombination happening. And so if you didn't update the training set, it would become less and less related to the candidates you're trying to predict every cycle. So every cycle you're doing recombination, you're moving the population further away from, the training set. So every time you update it, you're going to be kind of helping to keep that level of relationship high, so you have better accuracy. Okay. And then size of the training set is really important if you're trying to predict among a bunch of individuals that are not from the same family, but just from within a breeding program. you're going to have a pretty large population size. It all depends on the effective population size, which is, or basically the diversity of the program. How fast does LD decay in the program? The more diverse your population, the larger training set size you're going to need to achieve a good level of accuracy. And then there's some theory that says if your training set's really, really big, it will help maintain accuracy over cycles. So if you're able to get a really, really large training set, we should be able to do more cycles of selection without updating the model. That's what we expect based on theory. But I don't think we've got, in most crop examples that I've read, the training set sizes are nowhere even close to being like large enough to get to that point. So, and I should say though, the number of individuals that you really need depends on your population. So it depends how diverse it is. So key factor is the effective population size, which is really based on the history of the population. So has it been a very narrow, like few founders and few individuals mating every year? Or has it been really diverse and large number of founders and a lot of individuals mating every year? So that's sort of depends on the population history. And then, of course, heritability of the trait. So if the trait is of a lower heritability, you're going to need a bigger training set size as well. So here's actually a formula that's the expected accuracy based on the training set size and the population history. And I think a lot of people ask, like, how big does the training set need to be? It's like a very common question. And you can actually kind of solve that, answer that question here with this formula. So NP is the training set size. And then we've got here, this is heritability of the trait, also in the denominator. Then ME, which is the number of independent chromosome segments. And this relates to the diversity of the population. If you've got a really diverse population with, its population history has been with a lot of individuals mating every generation, large effective population size, then you're going to need more individuals, so a lot of independent chromosome segments. Also, ME also depends on L, which is the length of the genome in Morgan's. So the bigger the genome, the more individuals you need in the training set. And then on the other side of that, how many markers that is required, that's a common question. It depends again on the rate of linkage disequilibrium decay in the population. That's just a fancy way to say like how many chunks of genome are segregating in the population. So you need at least one marker per segregating segment. So if you can envision the genome as being like in these chunks of where there's the sequences that are linked, and then so you've got these groups of markers that are linked, that's the segregating segment. You need to have at least one marker. And of course, it's not as nice and neat as this. It's not like you have these discrete chunks that just segregate, but you get the basic idea. You need to have at least one marker for each segregating segment. So there's been some studies. I think this was a simulation study by Mulison 2009 that came up with a kind of some guidelines for how many that you need in how many markers you need. And they said for high accuracies, you need 10 times the effective population size times L, or L is the genome size in Morgan's. And then if you want to, to achieve just moderate to high accuracies, you can do just any times L. So say, for instance, effective population size. I haven't estimated it in our breeding program, actually, so I don't know what it is. But when I was at Erie in the rice breeding program, it was about 30 to 40. So if you say, and I don't know, what's the genome size of rice and Morgan's? I don't remember. Let's just say it's 10. So, so if the effective population size is 30 and L is equal to 10, then you just need 300 markers. Seems like really small. So if the effective population size is bigger, of course, then you need more markers. And so I'll just give a couple of examples where people have looked at what affects accuracy. So in this case, this is the effective marker density on accuracy, and this is in an oat population. So here, these are different traits. This one is beta glucan, days to heading, growth percentage, plant height, and yield. And we've got different numbers of markers here, ranging from 300 to 900. And look at the, this is the correlation between the phenotype and the breeding values. This isn't really accuracy, this is predictive ability, but it's related to accuracy. You can see for some traits, you really have a tiny effect. So for instance, beta glucan going from 300 to 900, there's a very tiny increase in accuracy. And then yield, it's a little bit more of a difference where you've got, okay, it starts out less than 0.2 and it goes to in between 0.25 and 0.3. So that's a pretty big difference. And as, yeah, this is marker density. Yeah, different traits also. So kind of some small differences again for plant height, it's very tiny differences. And so kind of depends, varies depending a little bit on the trait, but surprisingly for a lot of traits in this case, you can get pretty good accuracy with just a very tiny amount of markers. Like it doesn't have a huge influence. Here's another example. This is from Durham wheat, where they looked at, again, marker density, the effective marker density on accuracy. This was this trait's grain yield. This one's grain protein content. And you've got here, this is 100 markers. And then they tried 500, 1,000, 2,000, 3,000, and so on. So after about 1,000, you really don't see any improvement after that. So it pretty much, it increases, from 100 to 500 to 1,000. After 1,000, you don't see much of an improvement. Okay, and then the other factor that can affect accuracy, of course, is the choice of what model you use to train the model. So there's been a lot of research on like testing different models and trying to find models that are going to help improve accuracy. What the main conclusion of a lot of that research has been is that it kind of just depends on the genetic architecture of the trait. So if you're talking about a highly quantitative trait, it really fits the infinitesimal model. Ridge regression block, genomic block, they work pretty well. It's really hard to improve upon those models. If the genetic architecture is not fully, infinitesimal, but maybe you have some major effects, major loci segregating, there are some other models like Bayesian lasso, Bayes CPI. These are models that try to mimic, or if they try to fit those genetic architectures better. And so in that case, you can improve accuracy that way. And I include here the other extreme, like if you have a Mendelian trait, something completely Mendelian, then you're probably not going to be doing genomic selection, really. You should probably be doing marker assisted selection. Okay. So there's this interesting paper, Teslo et al. 2012, that I really like. They took a lot of data sets from that had been published before, either looking at GS models or something else, and they tested a bunch of different prediction models to those data sets. So it's like, it's not really a meta-analysis, it's like a reanalysis of all those existing studies, and tested a bunch of different models. And across a range of data sets, GBLUP and ridge regression BLUP, they worked really well. So on average, those were the models that were giving the best results across a wide range of data sets. And in some cases, you know, in some specific cases, there was an advantage to picking a different model. But I think one of their conclusions was, if you're going to test a bunch of models, make sure that the models that you're going to evaluate are really different from each other. Because if they're all really similar, you will get similar results. So test some models that are really different if you're going to go through that effort of testing a bunch of models. Okay. So now genomic BLUP, since it is such a useful model and, you know, widely applicable to many data sets, we'll, you know, explain a little bit more about it. So this is a mixed model. Y is the phenotype here. These are fixed effects. These are the random effects, which in our case, these are our genotype effects. And by genotypes, I mean, like, think about your lines or your individuals, the individuals that you're genotyping. Or I might just say line effects, for example. And then this is error. And then we say, so this U are our genotype effects. We also refer to these as BLUPs. So this is what we're trying to estimate. Those are our BLUPs, those are our estimated breeding values. We assume that these genotype effects, these BLUPs, are normally distributed with mean 0 and this covariance matrix. So this is, G is a relationship matrix based on marker genotypes. And this is just genetic variance. So we're saying that these genotypes are related according to this relationship matrix that we estimate using markers. And so the covariance among these breeding values is proportional to their relationship, their genetic relationship. And that makes a lot of sense because we know that individuals that are more closely related, they're going to resemble each other more. Their breeding values are going to be more similar. Their phenotypes are more similar. So the more things are related, the more similar they appear. So that's really all this is telling us. And this G, we can estimate it. The simplest way to estimate this G is take your marker matrix multiplied by the transpose of your marker matrix. This is a, marker-based covariance matrix. I think I have illustrated that somewhere. No, maybe not. But M, just to, because I don't think I have a picture of what M looks like. But M, this is your marker matrix. So if you've got, see, what does M look like? So you have your genotypes, your lines. These are on rows. And then you've got markers across the top. So, and then you will have some information. So at this individual, at this locus, you have its coded genotype. Let's say it's 1. This individual at this locus, its coded genotype is 0. Let's say this one, it's -1, and so on. And so you have this matrix that tells you for every line, and for every marker genotype, what is the genotype value. So that's what M looks like. And that allows you to generate this relationship matrix that tells you, how much you expect their breeding values to resemble each other. All right. I think, yeah, that's fine. I think these are advanced questions. We can discuss them later. So I'm gonna skip this slide. It's not that important. There's another relationship matrix. Actually, there's more than 2. There's different ways to generate these relationship matrices, but this is an important one. This is from Van Reyden 2008. And in this case, instead of it just being the marker matrix times its transpose, we first do something to that marker matrix. We don't just use the coded values. We subtract this value here 2 times pi minus 0.5. So this is, and pi is the frequency of the 1 allele. So this is a way, what this does, it gives more credit to rare alleles when calculating relationships. So if 2 individuals share a lot of rare alleles in common, it's going to give it's going to say that they're more related. Okay, and then the denominator here scales this relationship matrix. And what you end up with is the relationship matrix, it starts to look more like a relationship matrix that you get from pedigree. So normally, if you're using this version where it's just the marker matrix by its transpose, it doesn't look very similar. It doesn't look similar to a pedigree-based relationship matrix. This one definitely, it starts looking more similar to pedigree-based relationship matrix. So you can kind of use them more interchangeably. All right, and then ridge regression blob. Interestingly, it gives you exactly equivalent results to GBLUF, and it also uses a mixed model, but it just does it in a little bit different way. So now, it's kind of the same model, but so you have phenotype, fixed effects. Now our random effects are not our lines. They're not, but they're markers. So this Z matrix is actually going to be our marker genotypes that have been recoded. And this U, these are going to be blobs, but for each, like they're going to be marker effects actually. So this is going to be marker effects. And these are random effects. Yeah. So then with bridge regression block, we fit this model, we obtain marker effects, and then using those marker effects, we have to predict breeding value by multiplying the marker effects by the marker genotypes. So there's like a little bit of an extra step. We obtain the marker effects and then multiply it with the marker genotypes. Someone's asking if the genomic relationship matrix is same as the kinship matrix. So I think sometimes people have referred to them as the same thing, but the kinship matrix is actually based on pedigree. And it's, and actually to compute a relationship matrix, you have to multiply the kinship matrix by 2. So they are very similar, but a lot of people have referred to genomic relationship matrices as kinship matrices. So they're sort of a very loose use of the terminology. All right. Okay, so then how do you actually fit the model? So you kind of have 2 choices. You can fit the model all in one. So let's say you're using GBLUF. If you go back to GBLUF, let's zoom in here and I'll kind of erase my notes if I can. It's not letting me. I don't know. Okay, oops, that's not what I was trying to do. Okay, well, I'll just redo it then. So if you want to fit the model in just one step, then for one step. Then here you're going to put raw phenotypic data. And then here you're going to have all your fixed effects, all effects. These will be like environment, maybe trial, all these other effects that are related to how you conducted the phenotyping. And then here, it's just going to be your genotypes. So that's fitting it in one step. And then the other case would be if you have 2 steps. If you're going to do it in 2 steps, then it's the same model, but these are going to be like means, adjusted means. Then that takes in across all the environments. So you're already going to have to fit a different model that removes all the other effects, like environment effects, trial effects, things like that. And then you would just put adjusted means here. And then here would be just the intercept. And then here, genotypes lines. So most of the time, probably what's more more practical, more convenient is to fit the model in 2 steps. And that's because you've got a lot of factors to account for. Say you've got hundreds of different trials, and each one has some specific experimental design factors. Maybe there's a different blocking setup or spatial variation or something like that. So it's going to be more convenient most of the time to do some initial analysis of the data. Then what you're inputting is not raw data anymore. It's already analyzed data. They're adjusted means. And then in your fixed effects, maybe you've already accounted for all the fixed effects potentially, and then this would just be intercept. So this would be intercept or intercept. Hold on, what happened to my Zoom? Okay, so this is going to be intercept or any unaccounted for effects. So things that you did not account for in the earlier stage of analysis. And then if you do 2 steps, if you do analysis in 2 steps, you will have to account for heterogeneous error variances. And that just means that with your response, since you do have, you're dealing with adjusted means, you're dealing with, you're not dealing with raw data anymore. Each of those means potentially has a different standard error associated with it. And so we need to have a way to account for that. We can account for that using this, there's this residual covariance matrix, which is R times the error variance. And in the diagonal of R, we put 1 divided by a weight, where the weight is this reciprocal of the known error variances. So it's just 1 divided by standard error squared is RW. So that goes on the diagonal. So this is what goes on the diagonal of R. So it all sounds complicated, but when you do an example, it's easier than it looks. So, just goes in hand, implementing genomic selection. Actually, I think fitting the model sometimes is the easier part of the process, but just everything else leading up to that point is probably more, labor intensive and difficult. So first of all, just managing the data and using a database, or if you haven't already used one, getting all the data standardized and curated can be a big task, because there's probably going to be a lot of data that may not be actually meaningful. Maybe you don't want to use that for training the model, so that can be difficult. And then just accumulating all the data that you need. So you need to start genotyping everything being phenotyped, and a good way to start would just be to use all that available data to train the model. And then the third point is, it's a good idea if you really want to use GS to its full capacity. It's a good idea to avoid selection for your traits of interest without recording phenotypic data. So you don't want to select just on the fly. Ideally, you want to actually, if some trait is important to you, every time that you're doing selection, it should be on data that you've collected. You should be recording data more than just visually culling. So then fourth point is, if you want to get started, just begin genotyping and selecting prior to phenotyping. Just when you have enough genotypic and phenotypic data. So you can start, every year you genotype your candidates, you can use all that information to get started, just use phenotype and genotype together. Then once you have a big enough training set, you might get to the point where you can start just selecting based on a prediction. But in the beginning, you're probably going to want to do some rounds of just selecting based on both phenotype and genotype until you get enough data. Also, that's helpful to help, you can start doing some validation and check, are my accuracies good enough, get kind of the system going before actually like starting. Okay, now this is a quiz question. So true or false, the goal of genomic selection is to predict breeding value. Sorry, I gave you the answer. Oh my God. Well, I already gave you the answer there. Sorry. That's funny. So yeah, the goal of genomic selection is not to predict phenotypic value, it's to predict breeding value. See, like my mind can't even read that sentence. It resists. It can't. I won't even say the words. Funny. Okay, so I think we can take a little bit of a break and then get the exercise ready to go. I think it's in the box folder, so we can get that going. Okay, I think we can start again. So there was a question that I thought was interesting. If you've got a space plant F2 nursery, instead of selecting on single plants, should you record the yield? So I think that's a good point. If you're selecting on single plants in F2 nursery and you record yield, is that like a meaningful yield? Most of the time, I don't think it's going to be a meaningful yield evaluation on a single plant. I guess maybe in other species, like I don't know if you're dealing with trees or something, maybe it's maybe an individual tree would be a meaningful phenotype, but in most of our crops, that individual plant phenotype is not going to be a meaningful phenotype in a production environment, it's not going to translate to a production environment. So in that case, I don't advocate that you phenotype individual plants and use that for selection. I think if you're going to switch to a genomic selection strategy, you're probably not going to be doing selection in the earlier generations at all. Like you're going to want to be going quick. You're going to want to be reducing the breeding cycle time. So you're going to want to either you're going to be genotyping and predicting those individual plants, or you're going to be trying to get them, maybe self-pollinate them, generate something that you can phenotype reliably as quick as possible. So you either select based on a prediction or wait until you can get a really accurate phenotype and then phenotype those candidates. So I hope that kind of made sense. Yeah. And then, yeah, so somebody pointed out, I think in the case for animal breeding, phenotypes are on single animals, although a lot of the time you're looking at the progeny of those single animals. So, and then using all the relationship information. So it's a bit different. Okay. So this is kind of what we already talked about, but I wanted to discuss it again just because there's a nice article that points out how rapid cycle, selecting among non-inbred individuals can be really beneficial in helping accelerate rates of gain. So as we were saying, there's kind of two ways you can apply genomic selection. And one is that really rapid cycle, really short breeding cycle, select among non-inbred individuals. That will decrease breeding cycle time. It can be kind of challenging to implement because you have to have cheap genotyping, fast turnaround time. You have to have a big enough training set to where you can be able to do all those quick cycles without reducing accuracy so much, because it'll take a lot longer for you to update your prediction model with new data. And you're going to be turning those cycles really quick, but you're still going to have to be then generating lines and then generating data to update the model. So your model is going to be a little bit less up to date relative to your breeding population. So that's those are sort of the challenges associated with that. Now, genomic selection among lines, it's kind of easier to implement because you're really just generating lines, genotyping, phenotyping, whether or not you select before phenotyping or not depends on how accurate you think your model is. And there's less potential to accelerate gain from that. So this is just illustrating what rapid cycle selection, among non-inbred lines, and then among lines, it's just at a later stage. So we already went over this, so I won't go too much. And then you can do both. You can have genomic selection among, in a rapid cycle among non-inbred individuals and among lines. So at later stages of testing, you can still be implementing genomic selection there. Now, there was a paper in 2017 that proposed this. They call it a two-part strategy. It's just that having it's just a rapid cycle genomic selection in addition to genomic selection among lines. So you've got here, it's just where you're just crossing, generating new individuals, genotyping them, predicting them, selecting, crossing again. So you're just you're not doing any inbreeding at all in your population improvement. And then you take some plants from this pool, this population improvement cycle, and you put it into a product development cycle, which is just a selfing, putting it through all these different stages of testing and then identifying a variety. Also, from this pipeline, you're generating phenotypic data that you then use to update the prediction model. So that's what they mean by this two-part strategy. And here's some simulation results. So they look at what's the advantage of having this two-part strategy where you've got this really aggressive rapid cycle GS part, and then you've got your line development part. Is it, because one of the, you could just not have this population improvement cycle. You could just have product development with, let's say, at an early stage, relatively, let's say at this stage you could implement GS here and select parents, so you could still use GS to shorten the breeding cycle without having this. So what they wanted to look at is what would be the advantage of having this extra population improvement cycle. And so they see that here they have different assumptions. One, genotype by year is 0, which is not realistic. This one, where you've got some genotype by year interactions. And what they found is this rapid cycle plus genomic selection among lines is what gave the greatest rate of gain. Just genomic selection among lines was still good, but just not as, didn't give as good of a rate of gain. one big assumption in this study was that there was a conventional testing strategy assumed in all cases. So just to say, what does that mean? So they basically didn't make any changes to this testing strategy at all. They said it's going to be the same. We're not changing it. We're just going to test this two-part strategy versus not having the two-part strategy. If I was to go back and if I was the authors of this and I could like go back and try some other scenarios, I would probably say, okay, ask the question. Instead of investing in having this cycle here, what if you just invested that money into changing your testing a little bit? Because it's going from 500 to 50 to 10. That's pretty, that's pretty small, I would say. So instead of investing here, what if you invested the money here? What's that? You know, I don't know what the answer is, but it's an open question. I think that just kind of, you know, it's good to remember that, you know, sometimes it's, you know, this rapid cycle plus GS among lines, it's better than GS among lines, but that assumes this specific testing strategy and we didn't try anything else. So there's, you know, there's no way unless we actually test it and run the simulation, we won't know if perhaps a better strategy would be to not have this rapid cycle and just invest the resources in like a sparse testing genomic prediction strategy. You know, you just don't know. There's a lot of different things. There's a lot of different strategies that could be done, and it's kind of impossible to test every single one. So, okay. Let me see here. Yeah, so I think I'll just skip to this because I mentioned it. So I just threw out there like sparse testing without defining it, but so just to explain this a little better, sparse testing just means that instead of having like a conventional pipeline where you've got, you'll say you've got 500 lines and then you go to 100 and then you go to 50. And then let's say, you know, in a conventional strategy, you're going to have just, you know, you're going to have everything replicated maybe 2 or 3 times in a balanced data set. So everything goes to all the different locations. You could actually do, instead of that, you can do a sparse testing strategy. And so that's where you've got these different breeding stages. Let's say at an early stage, you have more individuals. You've got 3, in this case, I just said 310. Okay, and then in the next stage, you have fewer individuals, let's say 130, and then the last stage you have only 45. These are just numbers, you know, as an example. So in a sparse testing strategy, you would take, and so let's say you have 4 locations. So in sparse testing, you would actually divide up your germplasm, divide up the population. And now, instead of having all your individuals like in all the locations, you would just have, let's say in location 4, you just have 25% of your individuals from this breeding stage. In your location 3, you would have this other 25%. In location 2, you have this other quarter of the population. Location 1, you have this other set. And so you're dividing up your population so that each location that you're evaluating would get a sample of that. You're not having all individuals in all locations. They're sparsely tested. And then the same in the next breeding stage, it's the same principle, but now you're in each location, maybe you're testing 50%. of the lines in one of the locations, and then in the other location, another 50%, and so on. So it's a sample of each breeding stage. And then maybe in your final stage of testing, that's maybe where you would have everything in all locations. So you want to, probably get more data on those specifically to decide, what to release and so on. And so that's a concept with sparse testing. So instead of allocating, what that allows you to do is you end up generating more, it allows you to evaluate more lines, essentially. So you're getting, instead of being able to just test a few lines because like you replicate them all and put them all in every location, now you're able to test more lines, but you're not testing every line in every location. And that works really well with genomic selection, because what genomic selection allows you to do is analyze all that data together. It helps you be able to predict performance of every line in every location. So it sort of completes your data set, you could say. And so that's also really nice. It kind of opens that opportunity to use sparse testing more. Okay. I think I'll skip that actually. And then this is kind of another interesting paper that tried to look at this, answer this question: what's the optimal testing strategy if you're going to do genomic selection among lines? So is it better to do is it better to do more lines with less replication, so kind of a sparse testing strategy, so more lines, less replication, or should you try to do some lines that are, let's say you genotype some lines and not phenotype them? What's better? And here, this kind of like complicated graph is showing the what's the optimal, like what's the better strategy. Okay, so focus on so this is on the y-axis, it's the line plus the genotyping cost, and then on the x-axis, it's the number of individuals selected. So it's trying to say, okay, for what's the better strategy for these different parameters? Okay. So for instance, how do you read this? So if your cost of generating lines and genotyping is really high, so let's look at this, just look at this set of figures here. If your cost of generating line and genotyping is really, really high, the number of individuals that you genotype but not phenotype should be most of the time 0. Like you should be genotyping and phenotyping everything, except for when generating a line and genotyping is really, really small, and you're selecting a lot of, you want to, select a lot of individuals. So there's like very few scenarios in this case, when what they simulated, that it was actually worth it to genotype and not phenotype. So that, but there's, these few parameters, there's these few situations where, yes, it would make sense. Okay. And then, so let's see, this is number genotyped only. This is number phenotyped and genotyped. So this is this set of graphs, this one right here. So again, if it turns out sort of as genotyping gets more expensive, the number of lines and line generating lines and genotyping, the cost of those two things combined as that gets more expensive, it's the number of individuals that you phenotype and genotype is going to be getting smaller. That's the lighter color. And as it's cheaper, then you're going to want to more, there's this, the favorable scenario is to phenotype and genotype more lines. So, and then here, this is replication. So as generating a line and genotyping gets more expensive, it's better to replicate more. So if it's costing you more to generate that data, the line plus the data, marker data, it's going to be usually better to replicate more. So that creates a situation more favorable for replication. If it's really cheap to genotype and generate lines, then your better strategy will just be to generate more lines and have less replication. And in very few cases, like as genotyping gets really cheap and line generation really cheap, then it might make sense to just genotype only and not phenotype. So I think this kind of points out, like, it is really situation dependent. Even with this one data set, this is a simulation based on a single data set, just varying like cost assumptions, that the strategy of phenotyping and genotyping was all over the place depending on what costs were assumed. So it's, it can be very situation dependent. But I think overall, one thing that we should strive for is the cheaper that line generation plus genotyping gets, the better for genomic selection. Like the more gain we can actually get from genomic selection. So that should be kind of a no-brainer, but we want to try to get cheaper genotyping. See, I think we're almost done. Yeah. So I think a couple factors that I think are, things worth mentioning for implementing a genomic selection program, and maybe it might be common sense, but there's kind of two main factors, and that's the strategy, like how you're actually going to do it. Does the strategy make sense? Are you using the resources effectively, and really like taking advantage of genomic selection to its full potential? So that's all having to do with the strategy. And then the other part is, having to do with the operations. So are you able to actually get all the data together and, good data, meaningful data, and actually keep track of it all so that you can link it with marker data, and also being able to get these really quick turnaround times. I think that's really hard. So when it comes to strategy, what are some of the characteristics I think of a good strategy? First, I think it makes sense, if you're going to do genomic selection, you should be applying it to all the traits of interest. So it doesn't make sense to just do GS for one single trait, because once you genotype something, that cost is now already done. So let's say your primary trait, you say, oh, I want to do GS for yield. Well, why not other traits? I mean, you're already collecting the data, you already have the marker data, so you might as well use all of the information to its full potential. And then you should always be a good strategy. It should always involve phenotyping a large number of individuals from the breeding program. So lines, phenotyping lines, hybrids for model training and updating. So phenotyping is still, extremely important. It's, it's probably more important in a genomic selection strategy, I would argue, because you're using that data for multiple years, beyond. And in some conventional programs, maybe you're just using it within a year and then you don't use it anymore. But in a GS strategy, you're going to be, you really need good data because it's going to be used for a while. And then a good strategy would, ideally be using genomic prediction to reduce breeding cycle time. And you should be selecting parents based on GBVs. You should keep, try to allocate testing resources kind of in a appropriately, possibly using sparse testing. So, reducing replication when you can, try to use resources, most effectively. And then you should be having, cheap genotyping with enough markers. You don't have to have a huge number of markers, but just an adequate number. And then operational factors, I mean, there's just the key things like phenotyping, just as accurate as you can, given the resources available. And data management, definitely need a relational database with all the phenotypic and marker data. So you're not going to be able to do this if you don't have a way to use data across years. You're going to have to use and accumulate data for multiple years. So a database is going to be necessary. And then, good germplasm management. So being able to keep track of the identity of the individuals, like making sure the right, all the information is tracked, right seed in the right envelope. Because if you don't have that, you'll end up, you might have the most accurate selection in the world, but when you go to get the actual individual seed, it'll, it's not going to be the right one. So, if I think like ultimately, like it all comes down to like the very, like, these small details that are not trivial. And then fast turnaround time, inexpensive data, that's also critical. I think that's still a challenge for a lot of us, getting inexpensive marker data quickly. So that's the last of the slides, and let's see what other we have here. Someone's asking stuff. Does anyone have a question from here? Yeah.
So if you were working on like a long-lived species with like a long intergenerational time, is there a way to generate the training population without having to make a cross and grow out a set of projects for the first round selected?
So I think that's kind of risky, but I think you could do it. Like in my experience, like I did a experiment for my PhD, recurrent selection, and I tried using historical data in the first generation, and accuracy is like really, really, really low. So it was my conclusion after that, it's just not worth trying to do it until you have that training set. Yeah, any other questions? Software. I don't know. I don't have any product endorsement, but if you're working on wheat, you should use breed base and oats, I guess. Somebody's asking about if sparse testing, how do you fit the model? So I think I would be, if you're doing sparse testing, I think you'd be doing the same. It would still be a two-step, but what I would suggest is for every site, you can fit a model, get adjusted means. And then in the second step, you have site as a fixed effect and your genotypes as random. So you're not trying, you do two steps, but the first step doesn't account for all the effects. You do it by site and then you combine it. So that helps allow you to gain that connectivity from the marker data. Yeah. All right. So I think we can move on to the next part. I'll just pause for a minute. See, I'll just keep recording. Yeah, I think, wait, somebody's asking about data management in R or R Studio. So R Studio and R, like, that's great for analysis, but not very useful for data management itself. So I think you need a separate kind of database for that. Is Valerio, is Genovix an agro-based product? It's the new agro-base. Okay. All right. So we have a scenario here and some data. Oh, sorry. I'm getting confused. I have too many screens going here. I'm not sharing my screen. Hmm. Sign off on this one. All right. Continue. All right, so everybody, I hope you can access this file as well as there's an R data file that goes along with this. Should be in the box folder. So in this demonstration, we're going to show how can we use genomic selection for population improvement and line development, and then how to compare expected gain from genomic selection with gain from phenotypic selection, you know, to try to determine what would be better. Oh wait, I'm getting confused again. Okay. So imagine we have this breeding scenario. Okay. So let's say we have these 30 individuals, 30 founders, they haven't been selected yet. Then we intermate those, generate 100 F1s, and then generate 100 double haploids. Those double haploids, we phenotype and genotype them, and then we can predict a breeding value using all that information. And we're going to select the best 5, intermate them. It's okay. Okay. It's okay. So we select predict breeding value, select 5, intermate them, and then start again. Okay, so this is basic. Let's zoom out again here. So basic recurrent, kind of a recurrent selection program where selection is among double haploid lines. In this case, you know, the line development and the population improvement is all in one. We don't have like a separate rapid cycle line development. It's all in one. So then with this scenario, what I did is simulated that for 2 cycles and generated a data set. So it has 200 double haploid lines. So you'll see this in the data. We've got 200, 100 of them, which I've labeled them as group A. These are from the first cycle of selection. And the other 100 are, I've labeled them as group B. These are from the second cycle of selection. And then we have raw data on these 200 double haploids. I also have marginal means, so adjusted means from that. That's been provided to you as well. We have marker data. Marker data on everything that has been, used in the breeding process. Of course, we're only going to use the data on the 200 DHs because that's what we have phenotypic data for. Okay. So I'm going to get my RStudio here. I'm going to share. Oh, it doesn't let me share the desktop. Here it is. That's what I want to share. Okay. So now I can. So if you just go ahead and load that workspace, you can see these are all the objects that we've got. M, some marker data. Map tells us the position of the markers, which we don't really need, but it's there. Marginal means, these are, let's look at that. Okay, so it's giving, we have means for each individual. Okay. So we have the raw phenotypic data. So with the raw phenotypic data, we have for each individual, we have data from 4 different locations within a year. So to generate these marginal means, what I did was fit a model to that for each year, because the lines are actually different by year. So it's actually these are actually phenotypes. The marginal means are within a year. So we've got, that's why we've got here, there's still a year variable there. So we have some individuals from year 1 and some from year 2, because we've got one, each cycle, like the 100 DHs from the first cycle were tested in year 1. The 100 DHs from the second cycle were tested in year 2. What else can I explain? We also have true breeding value. So this tells us for every of our GIDs, what's the true breeding value. This is normally we don't have this information. This is just because it's simulated. So we actually know what the true breeding values are. What else? And then M is the marker data, so I'll show you that. So here's our marker data. This is just the first 10 rows and the first 10 columns. So you can see there it's coded -101, and let's check the dimensions. So in total, there's 430 individuals. So that includes the 30 founders, 100 F1s, and 100 DHs, and from cycle 1, and then again cycle 2, 100 F1s and 100 DHs. So that gives us 430 total individuals. And then markers, there's 1,200. Okay, so I kind of already, this is looking at the PDF again. So kind of what I just did, we have it summarized here. Looking at the true breeding values, which we know because we simulated them. So we simulate marker effects and then we calculate true breeding values. So we know for every individual, what's the true. And then marker data. So the individuals are across the rows and markers are across the columns. Okay. Let's see what we've got here. Say questions. Looks like, yes, 1 DH per 1 F1. That's correct. Okay, so here's our phenotypic data. This is raw data. So we've got for each individual that we've phenotyped, we have its phenotypic value, which we're calling P value. And then we have here in the data, you know, which location is this data from, what year. We also have this error and standard error. So in this case, this data is actually already been summarized. Like, let's say we have like 2 reps within a location. This would be the average of those 2 reps plus the error. So even though it's like raw phenotypic data, it's not actually like raw. Yeah, it's kind of already summarized. Okay. So we can look at the structure of the data using like str function. So str pheno, that gives us a summary, you know, of all the variables, what kind of variables they are. So in our phenotypic data, we have pheno GID is a factor. It has 200 levels. So for the 200 DHs, then location, we have 4 different levels of 4 locations. Year, we have 2 levels. So this is 2 years. And then phenotypic value is numeric, error is numeric, and standard error is numeric. And then we have a population variable, which is a character. We can, you know, we should probably convert this to a factor actually. Okay. So I'm actually gonna, now begins the demonstration part. So I'm gonna just put this to the side and go to R. Okay. So before we do anything, I'm going to create a new variable that's actually site or environment, you could say. So it's the combination of location and year. So this is actually this is what we're going to consider as our environment variable. It's a combination of location, year. So we're going to just add this to the phenotypic data. We'll do pheno $, environment, env. So that's going to create this new variable, env, which is the combination, which we're going to say is the combination of location and year. So we're going to paste, there's a function paste that allows us to combine those variables. So we'll do paste pheno $loc pheno $year. And then we're going to separate it by an underscore. I just chose, you know, arbitrarily, I'm going to separate it with an underscore. So that adds that variable, this environment variable here. Okay. So first thing, we're going to fit the mixed model. So we're going to use all the phenotypic data from the 200 DHs and then determine what are the best lines to use for the next cycle. Okay, so we're just going to assume that all the lines are unrelated. So we're not going to use any relationship information here yet. Okay. And then I have here, we have, 100 DHs tested in year 1 and the other 100 tested in year 2. So since we don't have anything in common across the 2 years. There's nothing connecting the 2 years. They're totally independent. So currently, there's no way, if we're not going to use marker data or anything, there's no way to actually combine all that data together to estimate across both sets of DHs, because there's going to be some confounding between the year and the population. So we don't have a way to separate that. So what I'm proposing to do here, what I do here is just fit the model to the year 2. So just using the data from year 2, we can evaluate, fit a model and identify the best individuals, ones with the highest flux. So we're going to use so we have to use LME4. So I'll do library. And then we're going to fit the model. Mod IID is LMER p-value +1 plus low plus. So this is going to, what we're saying is my phenotypic value is my response variable. 1 is the intercept. Sorry, this should be equals. Location is a fixed effect for locations. And then we have random effects for our GIDs. Those are just the the names of the DHs. And then my data is pheno. And then I'm gonna subset it so that I'm only looking at year 2. Oops. Okay, so we can run that. Whoops. Oh, sorry. Yeah. Yeah. Okay. Okay, so then we just want to get our random effects. We want to get the bluffs for our pheno GIDs so we get a bluff for every DH in our second population, the most recent population. So first we'll just save these bluffs in a new object. So I'll say result is ranf. So this is to get the random effects from the model. So we do ranf mod ID $sign pheno GID. So I just want to show you what that looks like. So this is that's what's in this object. It's just, these are just the best linear unbiased predictions, the bluffs, assuming the lines are not related. And here the row names correspond to the IDs of the DHs. And here are their bluffs. So I'm gonna change so the way that this is, the results are given to us. It's got, the IDs are in row names, and I'm gonna actually change it so that I have a data frame that, we have bluff, and then the IDs are kind of two separate variables. So that's what I'm gonna do here. I'm gonna do result data.frame phenol GID equals row names result and bluff Okay, so I just give these results back now in a data frame. Okay, so that's a little bit better now. Should say, I think I should call it bluff IID. Bluff IID. Okay. So now if I want to rank them, I can reorder it. I can say sort it by having the larger values at the top. So that's what this does. So we can do result order result bluff IID, and then decreasing equals true. One to 10. So what did I do? Interesting. There we go. So now I'm showing what are the top 10 individuals. So it's this individual 421, 410, 358, 430, so on. So if our goal was to just do selection based on our phenotypic data and recombine the top 10, this would be our selection process. We would take that most recent population, identify the best lines, and say, now, okay, we have the best, we can move on. And so this is kind of interesting. We can actually compare this with what's the true top 10. So remember, we have true breeding values. So for comparison, we can look, if we take our true breeding value data frame, we can again order it by breeding value with the highest true breeding values at the top. So that's what I did here in the demonstration PDF file here. And we can just check, how does that compare with what we actually selected based on phenotype. So yeah, definitely it's not the same. There's some things different. There's a few in common, but that does not mean that we failed. Just comparing those sets, those two groups, doesn't tell us our accuracy of selection. If we really want to check the accuracy of selection, we're looking at the correlation between the bluffs we just calculated and the true breeding values. So it's not, you're not going to get the exact same, you're not going to get the same set of 10 that are like the true best 10, but you're going to still get good lines. And that, it doesn't necessarily have to be the exact top 10. Okay. Somebody is asking some questions. So yeah, let's ask a couple of different questions. So here's a question. Somebody said, how do you estimate true breeding values for the data set? So first of all, we can never estimate true breeding value. That's a good question. It's only available if data is a simulated data set. So it's something like we're trying to always predict breeding value, but we never get true. We can never actually achieve, true breeding value. We didn't explain how to do the simulation just because that would take a little while. So I am not going to go into that. And then someone's asking, why is the location assumed to be fixed? Will the effective location, can it be random? So location can be random. It's actually usually most of the time should be random. But if you've got, like in our case, we only have 4 locations. So when you have something you would normally consider as a random effect, if you have just very few levels of that effect, it's usually better to treat it as fixed. So even though, let's say, in your thinking about what's the difference between a fixed and a random effect, a fixed effect or a random effect is something that is like a sample of all the possible, so your locations, for instance, it's a sample of all the possible locations where your lines could be tested in the same way. And so it definitely does fit, as a random effect, it can be considered as random. But in our case, we just have 4. So we're not, if you're going to fit something as random, you're going to be estimating a variance component. So if you've got just a few levels, like 4 or fewer levels, it's going to be better to actually treat that as fixed. So that's why we did that here. Okay. Somebody's asking that saying the true breeding value distribution looks skewed. I didn't really pay attention. I don't know. Okay. Sorry, I'm getting confused what screen I'm on. Okay. So there's something we can also compute that's very useful, and that's the reliability of the bluffs. So the reliability tells us how accurate, if we select based on these bluffs, what's our accuracy of selection going to be? So the reliability is actually the square of the selection accuracy. Remember, we talked about the breeder's equation and what's the selection accuracy. We can get the selection accuracy by computing reliability and taking the square root. So that's what we're going to do now. Okay, so we got SE bluff ID. So first we need to get the standard error of the bluff. I think there's a package we need first, actually. There's a package called ARM, library ARM. So you might have to install it if you don't have it installed. That allows us to get the standard errors of the bluffs. So we get standard error. There's a function, it's se.ranf. So we do se.ranf mod IID $sign signal PID. Okay. So that gives us the standard errors of the bluffs. Now we need to get the genetic variance. Okay, so first we need variance component. First, we need to get the variance components from the model. So I'm creating another object, which is vcomp. It's just going to be a data frame with our variance components. Data frame, data dot frame. And to get the variance components, we need this function varchor, varchor, and then mod ID. Okay, so now we can look what's in this vcomp object. We have our genotype, our genetic variance, and residual variance. So to get VG, we just do vcomp, subset it just to get this variance right here. So I'm going to say for the rows, it's going to be vcomp $group equals phenogid, and then VCOV. Okay, so that's genetic variance. I just noticed a mistake here in the demonstration. So before, the next step is actually, we need to estimate or calculate, sorry, the prediction error variance. So we have here the standard error. Now we'd have to square that. And that gives us prediction, this is a new term, prediction error variance. So PEV is equal to SE up IID squared. That's prediction error variance. So then the reliability is 1 minus PEV divided by VG. Okay. Same as our standard error squared. Yeah, it turns out that doesn't change a whole lot, but that's the reliability. Then if you want the accuracy, so I'm going to say selection accuracy is going to be the square root of this, of the reliability. So our selection accuracy, how accurate was that selection that we just did based on those blocks? It's 0.5. Okay. All right. So that's what we can do if we don't have marker data. So now we can go, what if we have marker data? How can we fit a model to this data? Let's see what we have. So what we're going to do, let's say we're going to use all the available data. We're going to use all the phenotypic data, all the marker data on all the 200 DHs. And from there, we can actually fit a model to everything, even though some are in different years, because now we can connect them by the marker data that, we have, even though we don't have the same lines across years, we do have genetic relationships across years. So we can, what we'll have to do, we'll have to take our marker data and then subset it so that, because right now the marker data has like 430 individuals and only 200 are actually phenotyped. So we'll take and subset it so that we have just the ones that have been phenotyped. Then we're going to create a relationship matrix. And then we'll create the design matrices for the mixed model. we can account for the heterogeneous errors here. There's sort of a way to pre-multiply by the inverse of the residual covariance matrix. And the trick to do that is actually pretty simple. You just take your design matrices and divide by the standard error. So it turns out to be relatively simple. And then we're going to fit the model and our mixed model with all the phenotypic data and our design matrices and our relationship matrix. And then we'll end up with blobs. These are going to be genomic blobs now. So these are GBVs. And they're for all the 200 DHs. And now we have these genomic blobs. We can compare it with the previous blobs we obtained. We can actually then combine, we can compare with true breeding values. So we'll do that. Okay. Let's start. Where do we start here? Okay, so we're starting at genomic blob. All right, so first thing, I'm going to do genomic blob. So we got to subset the marker data. I'm going to create M sub is M. And then I'm just going to take I'm going to use the pheno-GIDs that are in the phenotypic data to subset that marker data set. I'm doing here. Okay. Okay, so I'm just subsetting. Then we're going to create our marker-based relationship matrix. That's first we need the RR blob package for this. So we need library. Blob. And then we're going to do G sub. So this is our relationship matrix of that subset of individuals from the marker data. And we use this a-mat function, a.mat. And then the marker data M sub. So now we have our relationship matrix. I can show you what that looks like a little bit. Okay, so oops. Sorry, G sub, not G. Okay. So you can see we have all the individuals on rows, all the individuals on the columns, and every position in this matrix tells you the relationship between the combination of two individuals. So this one I just highlighted tells you the relationship coefficient between DH132 and DH131. Okay, oops. All right, so now we're going to create the design matrices. And I'll explain after we make them, I'll explain that a little bit better. So say design matrices. So X for our fixed effects. We're going to use a function called model.matrix, model.matrix, and then P value is our phenotypic value, tilde 1 plus environment. So in our case, we're treating environment as fixed, so it will go into this equation formula, and then data is pheno. Okay, so now let's look at what this design matrix actually looks like. Okay, so here, actually I should do view. See better. So this is what the design matrix looks like. Trying to find. Here we go. So, for example, what this tells you is for every phenotypic observation, kind of where did that phenotypic observation come from? Which environment did it come from? So, for example, this phenotypic observation at row 358, it came from environment 4_2. I think that was location 4, year 1. So that's where that came from. The intercept is just going to be 1s for everything. It's everything having the same, has the same intercept. So here, as another example, this observation number 468 comes from location 1, year 2. So it's kind of a matrix that just indicates where did that, what level of that factor does this value correspond to. Okay, so here's another. So for this one, 599, it's from location 2, year 2. And then, so you should have a 1 in one of these environment columns and a 0 in all the other ones, because it can only come from that one environment. So that's what the design matrix is. It's basically telling, where does that, how does that phenotypic value relate to the fixed effects that we're going to estimate. All right, now we're going to do the Z. This is our random effects design matrix. So for Z, we do again model.matrix P value tilde 0. So for the random effects, we don't have an intercept column. So in order to eliminate the intercept column, we put 0 there. So then it will not put an intercept column in the design matrix. So 0 plus, and then pheno_GID is our variable, and then data is pheno. So this one is harder to look at because it's going to have, I think 200 different columns. So every phenotypic observation, there's going to be a bunch of, it's going to say what genotype does it correspond to. So it's going to have all kinds of zeros, and then there'll be a 1 indicating what genotype it actually corresponds to. And then our Y is just the phenotypic data. So Y is pheno $P value for phenotypic value. Okay. Okay, so now those are our design matrices. If we look at let's look at our phenotypic data again. Real quick. So we have, for all of our phenotypic values here, we have the phenotypic value and the standard error. So let's suppose that these values that we have here actually came from like 2 replicates. Let's say it's an average of 2 reps. So we have a standard error here associated with that environment. So let's see, I can't remember how many different standard errors we have, but we can try to plot it so you can see better. Okay. So I'm just plotting the standard errors so you can see it looks like every environment has different standard errors. So we've got, there should be 8 here. 1, 2, 3, 4, 5, 6, 7, 8. Yeah, so every environment is having a different standard error. So we can actually account for that. And remember back in the slides, I talked about how if you're doing a 2-stage analysis and you've got, you're using a mean and a standard, and then you have a standard error, there's a way to incorporate that in the error covariance matrix. So there's this R matrix that tells you, where you can put, those weights according to your standard errors, and then that R multiplied by the error variance together, that's our residual covariance matrix. So we have so in the case of RR Bluff, the package that we're using, the way to actually do this is it's kind of a workaround, but we're pre-multiplying our design matrices. So this it's kind of equivalent to what to actually inputting, that R matrix while the model is being solved, but we're just kind of doing a pre-multiplication step. Okay, so I don't know why this is, it's not letting me here. Okay. So I'm gonna just say pre-multiplied design matrices. So we can do x2 is x divided by square root, or pheno, it's just the standard error column of the phenotypes. So yeah, we can just do a little bit of a shortcut there. Y2 is, sorry, x2, I already did that. Y2 is then y divided by pheno C, and then Z2 is Z divided by sorry, Z2. And I should emphasize, like, you only need to do this when you have these heterogeneous errors and that you know what those errors are. If you have data where, let's say, you didn't do any previous analysis, you're just putting in raw data, you wouldn't do this. It's only if you have known heterogeneous error variances that you have figured out from some previous stage of analysis. It can also be the case, like, say you may also have heterogeneous errors if you know, say for instance, you have well, it's still the same thing actually. I was going to say, if you replicated more times in one environment versus another.
I don't have an answer for that. Is there something else I can help with?
But it wouldn't really change this at all. So it's, so just think about it. If you know that you have heterogeneous errors and you have estimated them from a previous model, you would do this. Otherwise, you wouldn't. So even if there are some cases, or maybe you get some data and you know that some analysis has been done, but you don't actually have standard errors. In that case, you don't have any way to account. You have to just assume that errors are homogeneous because you have no other way to deal with it.
I'm not sure I understand.
That's funny. All right, so now we have everything ready. We can just fit the model. We can do our model is mixed. We're going to use mixed solve. So this is just a function that solves a mixed model. Y2 is the first argument. Then we need the random effect design matrix, Z2. Our relationship matrix, G sub, and then X2, our fixed effects design matrix. Okay. So that already solved, that model already fit. So now we can get the results. First, I'll show you what the results like that, what the output looks like. So if you do str mod, you've got here. Oops. So from the model, we've got VU. This is the additive genetic variance in the base population. So this is not among DHs. If we want it to be among DHs, we have to multiply this by 2. This is the error variance. These are the fixed effects. So for each of our 8 environments, we have a coefficient. Then we have U. These are our genomic estimated breeding values. And so what we're going to do is create a data frame where we have our PhenoGID names and their genomic blocks values. Okay. So let's do results, gblock data.frame, and then PhenoGID equals mod u. Oh, sorry, names, I think. Names mod u, yeah. And then what did I call it? Blup, gblup. Equals mod u. All right, so we've got the results. We can look at that. Oops. Oops. Okay. So here we got all the genomic estimated breeding values, or also in this case, because we use genomic blup, we can also refer to these as genomic blups. And then we've got the GIDs for associated with those. And now we can actually compare. So we're going to compare by merging our previous results from the model that we fit before, where we just fit without any relationship information. So we're going to do a merge. So combine results. And then we're going to do blup results, merge. And then these results we just estimated, result gblup. And then the previous results, which I think we just called it result RSLT. And then we're merging this by PhenoGID. Okay. So now let's look at that. So now we have the two. We've got our pheno-GIDs, our genomic bluffs, our bluff without relationship matrix, without any relationship information. Oh, I forgot to say all x equals true when I merged it. So we can do all x equals true. Because remember, our genomic bluffs, we actually were able to estimate for all 200, whereas our bluffs without relationship, our bluff IIDs, that's where we only had 100 of them. So there's going to be some that they're going to be missing. You'll see that. Okay. Yeah, so see, for instance, this individual 131, we have a genomic bluff, but we don't have a bluff IID because we only estimated that most recent population. Okay. So now probably what's even more interesting is if we want to compare these to the true breeding values. So this is really getting at like what's our actual accuracy of selection between these two different selection methods. So let's combine with the true breeding value. TBV. So we're going to do all BV is merge, bluff, result, TBV, and by pheno-GID. Okay. Okay. So now the moment of truth will be to actually correlate all these values and see what the accuracies are. Okay, so we're gonna do, I'm gonna do a correlation matrix with this, with these data that are in all BV. So I'm gonna correlate all the different breeding values with each other. But I'm gonna actually remove the missing values. So I'm making sure that I'm really correlating all using just the same set of individuals. So I'll do na.omit. And then I'll remove the first column because that's the pheno-GIDs. So that doesn't make sense to correlate anything with that. -1. And we get the correlation matrix. So here are this is actually telling us what's the selection accuracy now. So if we say, what's the selection accuracy if we just select based on our bluff, our IID bluff, so no marker data. And the selection accuracy turns out to be 0.18. So this bluff IID, that's correlation with the true breeding value. And then we have the genomic bluff estimate, what's the correlation between that and true breeding value? And it turns out to be 0.59. So definitely have a huge increase in accuracy compared to the previous model. Okay. So now with everything that we just did, we just looked at using, using gbluff, but using phenotype and genotype together for selection. So in that situation, like the scenario that we're interested in is selection after phenotyping. So that's what we just did. We just evaluated that. So of course, accuracy, we expect that to be pretty high. So we use all that previous information. We use marker data. We use the current phenotypic data that we just collected on our candidates. And we got this accuracy of like 0.6, like really high selection accuracy. So now we can ask the question, what if we did selection before phenotyping, actually really predicting those individuals, not phenotyping them, and then what's the accuracy of that prediction? So would that make sense? Okay. So basically all we need to do is take everything that we just did, but we need to exclude that population B. So we need to remove population B from that data set and then rerun it. So I could just show you, copy and paste and show you, but it's also here in the PDF. So I don't know if I really need to do that. It's the exact same process, just copy pasting. But here what we're doing is we're taking that phenotypic data and we're removing the rows of the phenotypic data that correspond to individuals in population B. That's what we're trying to predict. We're going to remove them. And then we create the design matrices, account for heterogeneous errors, fit the model, get the results, combine it with, this time we're just combining it with the true breeding value, and then say, what's the correlation between what we just generated, that G bluff we just generated, what's correlation with the true breeding value? That's accuracy. And it turns out we get that correlation is 0.45. So we went from about 0.6 to 0.45. And we save, I think we save about 1 year. So you can try to do the math and figure out, is it worth it? I think I did it already here. So if we look at basically all we need to do is look at that ratio of accuracy and breeding cycle time. So if our genomic selection before phenotyping, that accuracy, so I'm just gonna highlight here, we can zoom in. That accuracy was 0.452. We divide by the breeding cycle time, let's say it's 1.5 years. We have this 0.3 times, the other variables in the breeder's equation. And then we have, for the other strategy where we select after phenotyping, we have a higher accuracy, so 0.59. But if we look at that ratio, accuracy by time, we get 0.239. It's a gain by selecting before phenotyping in this situation. So now that's pretty much how you would, evaluate the different approaches. The only difference, of course, is you're not going to have true breeding value in your real data set. So you're going to need to have something else that is a substitute for the true breeding value. So something that is like a very good estimate of breeding value. And then the correlation that you get is what you're going to end up with is what we call like a predictive ability. And then you have to divide by the accuracy of that, what you used instead of true breeding value. And that will get you the best estimate of accuracy. So let's see what we got here. What did I miss here? It's like too many questions to answer. I don't know. Okay, well, here's one that is very, I think, critical to answer. So don't be confused in our phenotypic data. This p-value, it's not a p-value. It's not like a, it's a phenotypic value. It's just p-value, phenotypic. So it's not like a p-value. That's confusing. So it is our phenotypic value, actually. Somebody's asking, how do we interpret the result from the reliability? So the reliability is of a bluff. So we estimate bluffs, we can estimate, we get from each bluff, every time we estimate a bluff, we get a reliability of each bluff. And the reliability is going to be the selection accuracy squared. So if we were trying to determine like, if what's going to be our expected gain from selection of using that bluff, you can actually just look at the reliability, take the square root, and that should tell you the expected selection accuracy. I don't know, in this case, for some reason it's way off. I don't know. I think our reliability was, I think our selection accuracy based on reliability was like 0.5, but in the real case, it was like 0.18. So that's like way off. But I think the reason for that must be because we only phenotyped in like 1 year. So reliability looks high because we're not actually our genotypic values that we're estimating are actually genotype. It's like incorporating some of the G by E. So it's actually some genotype plus G by E effect. So it's looking like we have much higher accuracy. Yeah.